Good morning. We're going to go ahead and get started. If I can ask you all to start taking your seats. You got really quiet really fast. Thank you. Good morning, everyone. I'm Greg Daniel. I'm Deputy Center Director for the Duke Margolis Center for Health Policy. I'd like to welcome all of you to our second annual public conference on real-world data and real-world evidence. Today we have a lot of familiar faces in the room and on our panels, and we're going to really focus in and explore ways in which real-world data and real-world evidence can potentially support a range of stakeholder decisions with a particular focus on how real-world evidence can support regulatory decision-making and potentially map to regulatory standards. Well, Morgan, Jacqueline, and myself will provide a few opening presentations with regard to how we got to where we are today. The particular focus is because the 21st Century Cures Act and FDARA put forth a directive on FDA to explore how and when real-world evidence might be useful in regulatory decisions. So throughout our conference today, I'd like to do three major things. One is to take stock of where we've been in the last few years and how we've made tangible progress since our initial conversations around real-world evidence as part of the Cures talks approximately 2014. What have we done since then? And how can real-world evidence and real-world data map to regulatory standards? Second, discuss data and methods work that's still ongoing, both at the Duke Margolis Center as part of our collaborative, but with many other partner organizations across the country, and many are represented in the room today who also have been making progress on better ways to develop and use real-world data and real-world evidence, and trying to fill gaps in science and in policy in terms of using such evidence for decision-making. And then finally, looking forward to 2019. What should we expect to see in terms of progress between now and a year from now? We know that FDA will be coming out with framework and other activities, and we'd like to get a sense from all of you on what would be tangible progress to make sure that we continue to move this important field forward. So I'm going to spend a few minutes going through our agenda today. We'll first have a recap of the last few years from Morgan Romine, the research director here at the Duke Margolis Center, who oversees our real-world evidence and regulatory policy work. We'll then turn to Dr. Jacqueline Corrigan-Curry, director of the Office of Medical Policy and CDER at FDA, and Jacqueline will be providing some overview comments about FDA's current activities in this space. We'll jump into session one. We'll take a deep examination of whether and how fit for purpose real-world evidence can meet regulatory and statutory standards like substantial evidence and adequate and well-controlled clinical investigations. This is an area where Duke Margolis is actively developing a white paper, and we'll hear from some of the folks helping us to develop that in the discussion document, as well as FDA. In session two, we'll turn to key themes and recommendations found in a white paper that Duke Margolis released at the end of last week. This paper is orange. It's printed out on the registration table, but I'm told that it's sold out fast, and more copies are coming off the printer as we speak. This paper focuses on characterizing the quality and relevancy of real-world data sources for regulatory use. In that session, we'll be joined by a great lineup of working group members and outside experts to discuss the data side of the real-world evidence equation. In session three, following lunch, we'll have some updates from a number of organizations pursuing real-world data and real-world evidence projects to help inform their use in clinical practice, research, and potential regulatory submissions. And then finally, in session four, we'll close with some forward-looking discussion on shared goals and next steps and what we hope to have accomplished in a year from now. As I mentioned, we have a stellar lineup. I'm excited to dig in. And before we do, though, I'd like to spend a moment thanking our Duke Margolis Real-World Evidence Collaborative, who made this conference and the multi-stakeholder work possible. Since last year, we've been working with the organizations you see here to develop white papers, hold expert workshops, argue, get into really good discussions about an important topic. And this is work that we believe is best undertaken together. No single organization could really move this forward and get to some great discussion and consensus around some of these topics. The work is not funded by the FDA, and I do want to make sure to note that while FDA experts take part in our collaborative meetings as observers, the work products and views expressed by us at the Duke Margolis Center and the work products from our collaborative partners are not those of the agency. This work has and will continue to be open to participation and input from other organizations and experts, so we look forward to working with all of you in the room and online as we move projects forward into 2019. One last thing before we get going, I have a few housekeeping comments to take note of. This morning is being live webcast, so while we will not be formally reporting out comments by specific speakers or participants, everything said here should be considered on the record. Knowing the group we have assembled, I doubt that many of you will hold back. And I would like to take the moment to welcome all of you joining us online. We have ample time for audience comments and discussion, so please, if you're in the room, we have mics on both sides of the room. If you're joining us online, you can submit questions by tweeting us at Duke Margolis, that's our handle, or emailing questions to margolis.rwe@duke.edu. And our staff will try to get to those questions and run them up to me as I'm moderating these sessions throughout the day, and hopefully I'll get to those questions as well. As you'll note from the agenda, we have built-in breaks throughout the day. If you're here, we have coffee, tea, and drinks outside of the room for your convenience. Lastly, lunch will be on your own, but there are a number of close-by fast restaurants in the area. With that, I'd like to turn things over to Morgan to provide a bit of a background presentation.
All right. Well, thanks, Greg, and allow me to echo the welcome today. So. We always joke that there are a lot of familiar faces in the room, but I think the extent to which I see some of you in my sleep these days is a little bit of cause for concern. But we wanted to take a few minutes to talk a little bit about where we've come since 2014, right? So acknowledging that we have been using these types of data and evidence for a range of issues and challenges and decisions for a long time now. But the regulatory component did start to come into better focus in 2014 with initial conversations around the 21st Century Cures Act. And I don't want to belabor the act itself or PDUFA 6 too much. I think we're all pretty familiar with the language there and the mandates. But this is when a lot of the people in this room started to talk a lot more about how and when to potentially use these data and evidence for regulatory purposes. As you can see, it established a general roadmap for how Congress envisioned this process going, starting with the legislative mandates, working through public convenings and meetings to really solicit feedback and guidance from you all externally, a framework that will be out later this year from FDA, pilots over the next couple of years, and then guidance with a goal of 2021 for those guidance documents. So we're all pretty familiar with this landscape at this point and a few years in. But there were remaining challenges coming out of passage in 2016, one of which was terminology itself. So for those of you who've been on this ride this entire time, some of you might remember for a long time we were trying to make the term evidence from clinical experience happen, not even use real-world data or real-world evidence. We couldn't define that either. So that was scrapped in the legislative process. And eventually we did settle on real-world data and real-world evidence. But still, these are terms that over the last few years we've understood people are not using in the same way. And the first big hurdle was just making sure we were all on the same page. These are the definitions that we now use pretty much commonly across the board, both in the Duke Margolis papers, in a lot of the FDA writing on this topic. So real-world data are data relating to patient health status and or the delivery of health care routinely collected from a variety of sources. You can see those sources listed here. And then it's real-world data that becomes real-world evidence through the application of analytical methods. Specifically for regulatory applications, it can be further defined as clinical evidence regarding the use and potential benefits or risks of a medical product derived from analysis of RWD. Now, we always want to caveat here, too, that RWE can include randomization and non-randomized design, so making sure that, again, we have that level set. I think we're getting there. It seems as though most people are using these terminologies correctly and consistently. I'll leave it to Greg and Christina and the RWD panel to get into the discussion about types versus source versus where the data comes from, because there's some further work that probably needs to be done there. But we've particularly made progress here on just making sure that everybody's using the same terms. Now, a lot of that has been accomplished through pretty significant convening activities that have happened over the last couple of years. FDA has engaged directly through their cooperative agreement with us at Duke Margolis and through funding at the National Academies to hold public meetings over the last few years. And then other groups, both domestically and increasingly internationally, are having their own efforts and meetings to further explore potential regulatory use. So just this year alone, the academies, us at Duke Margolis, Friends of Cancer Research, the Academy of Medical Sciences in the UK, the Bipartisan Policy Center, Citi, there are tons of people acting in this space and holding ongoing discussions. And I'm sure I'll see some of you again later this month in New York for the New York Academies meeting, and then in San Francisco, a plug for the DIA RWE conference in November. But plenty to keep us all busy and going to RWE conferences, where I can also put in a shameless plug to come work with me and help me divide and conquer a little bit. We are hiring at Duke Margolis. And then we do have plenty of publications as well. So, FDA has made repeated statements in the press, in JAMA, in NEJM, and other places, laying out their thinking, helping with that terminology reset, and helping to kind of guide all of us along as we go. There's also been a number of frameworks and proceedings from the National Academies, the paper that we released last fall under a cooperative agreement with the agency, and then also increasingly other areas that eventually do need to be folded into this broader RWE umbrella, like mHealth and mobile technologies and how we're using patient-generated health data. It's a crude metric, but if you play around with your search on PubMed just a bit, you do get almost 3,000 hits since January 2016 with the term real world in the title. Now, that's representative of a lot of different types of work and opinions and endpoints development and different things, but there's a lot going on in this space, and we need to do a lot more. Now, if you've been coming to a lot of the meetings, you've also heard a lot about things like Salford Lung Study, Adaptable, the TVT registry. My main point with this slide is that this is still where we do need a lot of work. So since 2014, since 2016 with passage of the Act, we do still need more proof of concept work across registries, pilot studies. And importantly, our call from Duke Margolis continues to be that should be transparent and it should be additive to the overall learning experience as people work through some of these methods and data issues. So where are we today? That's one of the things that we want to have as a through line for the rest of the day. Are we making tangible progress? Are our conversations productively moving forward? Again, we've been to a lot of meetings like this, and are we actually getting anywhere? And what do we want to have accomplished this time next year? With a framework coming out later this year, with an annualized Duke Margolis conference that will be next October, what are some of the goals that we hope that we can make across data development, methods development, and some of these pilot and demonstration projects? So a tall order maybe, but I think we can eventually get there. And with that, I'm going to turn it over to Dr. Jacqueline Corrigan-Kurai, who's the Director of the Office of Medical Policy at CDER at FDA, for an overview of what the agency is working on.
Thanks, Morgan, and to Duke for allowing us to participate in this meeting. It's really a pleasure, and you'll hear from some of my colleagues as we go forward. So I'm just going to give you a quick update, a little bit of an overview of where we are and some of the things that we're thinking about. I think Morgan has really covered this very nicely, so I'm going to skip this slide. It's the 21st Century Cures, what we're expected to do, and of course our framework is due at the end of the year. So, incorporating real-world evidence to evidence generation, there's just many factors. I mean, with any kind of questions, when you're going from the question to the design, the highest level, there's a number of factors that don't change, regardless of whether this is real-world evidence or not. But of course, what we're adding now is this real-world data. And really, what does it do in terms of changing the types of questions, how we might answer them, and how do we think about the relevancy, the validation, the quality assurance and control? And of course, we'd already been looking at some of this in guidance that was done in 2013, so we have a base to build upon. But I think certainly it really is the data that we still need to focus a lot on. I mean, we have no lack of data, but we still have a lack of our understanding about the quality and what the data can tell us. And so at the very beginning, is it going to be captured? What we're looking for, is it captured in routine clinical practice? Is it consistently measured? How do we measure that it's consistent? What about the frequency of the assessment, the data collected from unique subjects of patients, or is it representative? The quality is a huge question for us. Is it possible to capture in multiple databases so we can have more confidence as patients move between providers and systems? And how much data is missing and is random? And if there are gaps, how we can fill them? And I know the paper that was released attempts to sort of articulate some of these matters. I think another big question and an area that we are going to focus on is understanding the sort of what we call data curation. So even the most structured of data certainly is not clean data and ready for research use. And as the quote from the Pink Sheet notices, even a data element such as a lab, a very simple lab, can be represented in many different ways. You know, we need to understand the relationships between what we're picking out in this real-world data in terms of endpoints, and how does it relate to the endpoints that we understand from a regulatory perspective. So research such as the one on the left from the cardiologist, trying to look at what about if we look at sort of MI and stroke and bleeding and claims, what's the relationship of that to what we have traditionally been doing with physician adjudication? And of course, some of the work that was done this summer by Friends of Cancer Research, looking in non-small cell lung cancer and the checkpoint inhibitors and trying to look at the correlation, which they found some correlation between real-world endpoints, and they were looking at the correlation to survival. And interestingly, they also found, at least in this field, that the results of our clinical trials were quite generalizable to the broader patient if you look at the issue of overall survival. So we're looking at all sorts of designs. We're looking from the very beginning of how you really start using this data to improve efficiency, and I know our partners at Citi are working on that aspect. Can we get folks to start thinking about more pragmatic and integration of trials? What about our single-arm studies, which we will always have? What can real-world data add to that external control? And when might sort of more observational study designs be appropriate and greater reliance on real-world data? So, you know, most of you know that we have published and sort of, you know, are supportive of randomization and really think it's important to think about randomization as a way to minimize bias. And of course, I don't think we're alone, and this is another recent interview, just talking about, we're not talking about we're only going to randomize in the pre-space and then we're going to stop randomization. We need to continue to think of randomization and real-world evidence as compatible. And so we think there's great opportunity into trying to design regulatory trials with pragmatic features. And that would be identification of relevant questions that practitioners and patients will find meaningful and participate in, selection of interventions that can be really integrated into clinical practice. For studies of approved drugs, streamlined data collection, as we've already provided guidance on. Integration of clinical data across healthcare systems is going to be important if we're really going to realize the potential of this. And indeed, you know, perhaps utilizing mobile technologies to fill in the gaps, including the capture of patient-reported outcomes. Because if we only rely on clinical records and claims, we're not going to get that patient voice directly. And so we know that many trials can have pragmatic elements while maintaining rigorous standards for data collection and assessment. And then what about randomized studies? Where are they being used now? Well, certainly in oncology and rare diseases, non-randomized studies continue to be used where there's small patients or ethical considerations. And of course, as we said, there may be great robustness that can be added to those external controls with you have greater data sets, you can match the patients to your trial. And then it can contribute to post-marketing commitments, as was done in 2017 for a rare disease. This is for cystic for Kalydeco, when they expanded the indications from 10 to 33, based in part in vitro data. The post-marketing commitment is an observational study to further understand clinical response in those patients. And of course, what made this possible was a robust registry of the U.S. Cystic Fibrosis Foundation that captured the endpoints that we needed to have captured. But we don't always see that we're moving and taking steps into other areas that are not rare disease. And our colleagues at CBER recently updated a label looking at long-term efficacy for the herpes zoster vaccine. So I'm not going to read this trial, but as you can see, the pre-approval was sort of your traditional RCT. But in the 50 to 59 group, they didn't have that much follow-up in part due to medical practice, changes in medical practice. So they were able to use a prospective observational study to gather more data about the long-term effectiveness, and that study continues to go on. But I know we're interested about additional opportunities, and this will be what's on the first panel agenda. And when we start to think about this, there's just a number of questions we're still working through. You know, would the study build on existing evidence of effectiveness? Are there potential advantages to a non-randomized design that perhaps we couldn't do in an RCT? Do we need empirical equipoise? And that's a term I sort of took from the comparative paper by Dr. Walkers and Schneeweiss and all, which in our view is that physicians are choosing between two different therapies if we're going to compare them, but there's not a huge bias or channeling towards one that could easily influence the patient and the physician assessment of that therapy. Are there relevant covariates that are captured, and can they be controlled? And are the results consistent across different databases and with existing effectiveness evidence? And can we assess that? Do we know what the unmeasured bias might have done to the results? And I'm sure there's other questions. But certainly, as we move in this, we need the same sort of transparency that we've had around clinical trials. And I think the ISPOR-ISPI recommendations are certainly a great starting point and very important for us to think about. And I'd like to just close by saying, you know, we're all in this together. We're all sharing a common goal. We want to meet the patient's needs. And as we do that, I think it's important to continue to be transparent in what we're doing, have the utmost protection of privacy, and then ensure our conclusions about a drug's effectiveness is based on the appropriate evidence. And we have had a lot of meetings, and we are, I think, moving forward. And I understand that for some, it may not be as fast as we think, but I think, you know, this is a quote back from '97 when the FDA Modernization Act was implemented, and I think the same is true. There's this just natural tension as we start to think about innovating and breaking through how we develop pharmaceutical drugs and medical practices and protecting the public by only approving safe and efficacious projects. So we move forward, but with scientific rigor. And with that, I'd like to thank every one of my colleagues, and I look forward to discussions today.
Thanks, Jacqueline. That's a great way to open up this this event, as well as the first panel. Really have given us a lot of things to consider, and thanks to Morgan for that great background. So I'd like to go ahead and jump right into session one. I'll ask our panelists to join me up on stage. We'll have Mark Berger, who's retired, but now joining us today as part of ISPOR. Nancy Dreyer is Chief Scientific Officer and Global Chief Scientific Affairs at IQVIA, Real World and Analytic Solutions. Stacy Holdsworth is Senior Advisor, Global Regulatory Affairs at Eli Lilly and Company. Dick Wilkie is Chief Scientific Officer, also at ISPOR. And also Peter Stein is Deputy Director of the Office of New Drugs at CDER FDA. And before we, no, please. I can stand. It's a big panel. Okay, so I'm going to kick off this panel with a few opening topics to really get into the point of this session. I'd first like to thank many of the collaborative working group members who have spent many meetings with us discussing and debating the specific issues of two things. How do you improve the credibility of observational designs to potentially support regulatory decisions? And then also, how can we think about the regulatory standards and mapping observational studies to regulatory standards? And I'm going to explain why we're focused a bit more on observational studies in just a moment. But to reorient those of you who are relatively new to the discussion, but many of you know this, the recent legislation by 21st Century Cures Act and the PDUFA 6 provisions under FDARA both sort of align pretty nicely and direct the FDA to explore the uses of real-world data and real-world evidence for regulatory decisions by doing things like public events and coming out with a framework and doing pilot studies. 21st Century Cures gets a bit more specific and specifies the FDA to establish a program to evaluate the potential use of real-world evidence to support the approval of a new indication for an already approved drug, or to help support and satisfy post-approval study requirements. And along the lines is to issue a draft framework this year and to work toward guidance by the end of 2021. So I want to spend a little bit of time talking about the value of real-world evidence for estimating treatment effects. There's a lot of reasons for thinking about how real-world evidence might support regulatory decisions without saying that they're going to replace traditional clinical trials, which will continue to be the gold standard for drug development. But real-world evidence does offer some opportunities versus traditional randomized controlled trials, and this includes both observational and randomized designs, as Jacqueline included in her presentation, that real-world evidence can offer the opportunity to generate evidence that's on a broader population, a broader uses that are more reflective of typical routine practice. Real-world evidence can include effects on longer-term endpoints or endpoints that are potentially more relevant to patients and caregivers. Not to say that you can't necessarily measure these in traditional randomized controlled trial, but real-world evidence gives the opportunity to more routinely collect these data and use them in evidence development. And then specifically focusing in on observational designs, while there are, and we're going to discuss, questions and concerns about validity and reliability when you're not randomizing, observational studies do offer the opportunity to develop robust, high-quality evidence using great data that we're going to talk about in the next session, how do you know it's great data, and sophisticated methods for getting to potentially causal effect estimates when randomization is not feasible. Observational designs and big data sets can enable, as I mentioned, longer-term follow-up and better, maybe outcomes that are more relevant to patients, as well as potentially to be developed, as evidence to be developed in a more cost-effective ways. So then, sort of narrowing down, okay, so then what could be the value specifically in the use of real-world evidence as it's stipulated in the 21st Century Cures Act, specifically around new indications and things like label revisions, subsequent regulatory decisions for a product that's already on the market. Well, first of all, by providing a path or some clarity for sponsors, this could potentially accelerate the development of high-quality real-world evidence, real-world data earlier on in the product life cycle than is routinely done today, and that could accelerate the availability of this information that might be more relevant to not just patients and providers, but also potentially payers and others that are making important healthcare decisions. But what's most important, I think, within that is that the product labeling, the package insert, or the labeling that gets approved is often used by providers and patients. You go online and you Google a drug and you find information if you're a patient that's going to go on that drug. A lot of the information that comes up is from the labeling. And by enabling real-world evidence to support new information that goes into the label, can potentially help patients and caregivers and providers understand more about how that product can work for these patients. Okay. However, the ultimate regulatory acceptability does depend on how robust these studies can be, how great the data can be, like we're going to talk about in the next session, and how can we trust and make sure that the studies that we're using to enable information to go into labeling is evidence that we can really trust. Last year, our first white paper, which is out there, the blue one, put forth this is small print for you all, but I think you know what it says. A framework generally to think about, okay, so what are the factors that go into, maybe as a sponsor that's considering developing a package of real-world evidence that might get submitted to FDA? What are some of the factors to consider on whether or not real-world evidence would bring credible information to the question at hand? On the left, in the green and the blue, are sort of like what the first two fundamental factors are, what is the regulatory question that's being sought? A new indication, new information in the label for the existing indication, a new population, all kinds of, questions might, give rise to, okay, do we think real-world evidence might bring important information to bear on this question? And that is in relation to the clinical context, which is, what do we already know about the disease? What do we know about the treatment options for this disease, as well as this drug under consideration? Is there clinical equipoise? What is the totality of evidence already? What do we know? What do we don't know? And what do we think? And why do we think real-world evidence might actually bring credible information to bear? So. Okay, so those two questions then, okay, so maybe real-world evidence can be useful, but only if we take a look at, are the data available? Are the data that are available high quality and highly relevant? And we actually define that later on today, as well as the methods. What are the best methods? Is it feasible to randomize? Is it not feasible? Is there a really good reason for doing a prospective observational study versus a retrospective study? And regardless of that design, are the analytic approaches that we're using to control for bias, observed and unobserved potentially, are those appropriate? And are we getting effect estimates that we can really trust? So all of these sort of factors come together when we're trying to consider, okay, can the real-world evidence be fit for purpose or fit for regulatory purpose? So companies, as they're thinking, go through some of these factors to develop the real-world evidence. But ultimately, the overarching question is, can we meet the regulatory standards with credible and robust RWE? So turning to those standards, 21 CFR 314 does a pretty good job of sort of describing some of these things. We know that FDA has a substantial evidence standard for making decisions, and the studies that contribute to substantial evidence must be adequate and well controlled. And this is important. So if we're thinking about, okay, so could real-world evidence support regulatory decisions, then the question is, can real-world evidence be considered adequate and well-controlled at the individual study level? And can the totality of evidence that goes into that package meet the substantial evidence threshold? So I think sort of the first part of this question is adequate and well-controlled, because that generally applies to an individual investigation. And 21 CFR 314.126 puts forth some quite extensive descriptions of what adequate and well-controlled includes. So characteristics of an individual study. First, a protocol and results report should contain or must contain clear objective statement and summary of the proposed methods and analyses. The use of valid comparisons with a control, and sets forth sort of placebo control, concurrent dose control, active comparator, and even goes through historical controls. A method for selecting patients that adequately assures that they have the disease. Treatment assignment methods that minimize bias and ensure comparability between arms. Ordinarily, this is randomization, but not always. Measures to minimize subject, observer, and analyst bias, such as blinding. Well-defined and reliable methods for assessing patient response, and adequate analytic plans for assessing the effects of the drug. So that brings me to, you all are going to answer these questions more generally, but I want to, so why have I focused so much on observational studies since real-world evidence does include randomized studies as well? And quite simply, it's that, for randomized, and Jacqueline put a lot of good examples forward on, real-world evidence that does include pragmatic trials and pragmatic designs and the ability to keep randomization in the clinical setting, which is critically important to do. And there are still questions about, can real-world data adequately support those kinds of trials? How do we think about the collection and use of data to meet the substantial evidence standard with randomized, pragmatic real-world evidence studies? But then if you think about sort of observational real-world evidence, there are a lot more questions on whether or not observational studies could, in an individual study, meet adequate and well-controlled, and together, what that sort of evidence package that gets submitted to the FDA that potentially could include one or more observational studies, but it also could include randomized studies as well. How does that package get to substantial evidence? So a lot of questions around that I'm going to direct to this panel and to all of you, particularly this morning, but I think these questions will come up throughout the day. So first is, for observational real-world evidence, we've said, when randomization is infeasible. So when is that? Is that the only time we would expect that observational real-world evidence could be supportive of a regulatory decision? And that's my sort of first question to the panel, is around what factors would lead the decision to pursue observational studies in the regulatory setting. Then I want to get to, on the left side of this, adequate and well-controlled. How do we know if an observational study can be considered adequate and well-controlled? What should they look like? Appropriate comparisons, balanced groups, adequate control for observed biases. What if you think you have unobserved biases? What are some of the things that we can say, well, if we could just do this, then we would know that this observational study is adequate and well-controlled. Difficult as that question may be, the other question is with regard to then substantial evidence. If an evidence package includes adequate and well-controlled observational studies, assuming we can do those, what factors would go into substantial evidence? So just because you have an adequate and well-controlled observational study, or two of them, doesn't necessarily mean that that's going to get you to substantial evidence. And so I don't expect you all to answer this, but give thoughts on sort of, so what would the factors be that would go into whether or not these studies would contribute to substantial evidence? Is it only if the treatment effect size is really big, then you would be more confident that this would be substantial evidence? Is it only or with multiple studies that demonstrate consistency across a number of observational studies? Does it depend on the strength of the relevant prior evidence? If you've had, if the randomized trials that were submitted for this drug to get on the market the first time, if it's the same population or you have estimates on the outcome that you're looking at now, does that give the ability to get to the substantial evidence standard for this? And sort of regulatory flexibility that we see in the areas of rare disease and severe high unmet need, does that factor in? As well as the specific regulatory question. So with that, I'm going to go ahead and turn to our panelists, which I have my questions right here. Okay, are you guys ready? All right, really easy question. So the first one, let's go back to the first one, which is around, why would you even pursue an observational design in the regulatory setting, particularly for like new indications and label revision? So I might go to, this is different than what I told you all in email. I'm just trying to keep you up on the, okay?
Keeping us on.
Yeah, that's right. So I'm going to go to Stacy first, if that's okay. Did you give some thoughts from, you come from the sponsor perspective on, what are some of these factors that might go into that?
Sure. Well, thank you, Greg, for the opportunity to be on the panel today, and thank you to Duke Margolis for convening this important discussion about real-world evidence and regulatory decision-making. The historical precedent of relying on traditional randomized clinical trials to provide the confirmatory evidence of efficacy has really put us in a place where FDA can make decisions based on a very high degree of internal validity. There is always going to be a need for these randomized clinical trials. But the external validity that's contributed by studies that are conducted in real-world settings has the benefit of greater generalizability to the populations that are closely going to match the patients that are actually going to use the medicine. These real-world studies are more reflective to the broad patient populations, the settings of care, the outcomes, especially longer-term outcomes, measured in actual clinical practice. So I don't think it's going to be a question of one versus the other in the future, but rather some combination of the two that should change how we think about total evidence packages in the future. In my mind, the ideal development plan would combine a number of study designs in various drug data types to maximize the understanding about a drug's benefits and risks. And with proper design and analytics and demonstrated reproducibility in independent data sets, observational studies can yield robust evidence that can be considered as part of the substantial evidence package.
Great, thank you. Mark, what do you think in terms of just sort of why pursue an observational design in the regulatory setting?
Well, you've already hit some of the major points. Clearly sometimes it's infeasible, and if it's infeasible, then you do use what you can. And then there are the issues about long-term follow-up is required. I mean, not everyone wants to conduct a clinical trial that's going to go 7 or 8 or 9 years. So, and the cost of that would be quite high. And then there's the regulatory context. If a treatment has a very strong safety profile, it's a really safe product, so that the potential for harm is really low, it becomes easier then to do an observational study to look at potentially label revisions, and even potentially down the line, although I don't think it's going to be anytime soon, a new indication. Clearly, it has to do with how much uncertainty can be tolerated and the proximity of the thing that you're trying to evaluate to existing claims. However, I could imagine that there will be, especially with the way that therapeutics are evolving, there might be off-label uses of a therapeutic that have such a large effect size that it becomes obvious to everyone that it probably has a real therapeutic benefit. And the question then is, do you then conduct a randomized trial anyway? And I think that there is always going to be discussion around this, because in the past, when things have become obvious sometimes, they really weren't obvious. So like autologous bone marrow transplantation for salvage therapy for breast cancer, where everybody thought it was obvious, it was valuable, but in fact, when the RCTs were done, the risk-benefit was not so great. Having said that, I don't think that's going to be the situation going forward, particularly as we have targeted and molecular and biomarker-directed therapies. I can imagine that there will be therapies that will come out which will surprise us with unexpected benefits. And when we get these kinds of unexpected benefits, if one has done rigorous multiple observational studies where one has pre-specified a protocol and a data analysis plan, where one has had adequate transparency so that others can go back and reproduce what your analysis was, and that one has used all of the rigorous methods to deal with confounding, and we get all of the results pointing in the same direction, and the effect size is very large, it won't make a lot of sense at that time for the FDA to say, no, yes, but you need to do a randomized controlled clinical trial, because unfortunately, the world is going to move on without them. Clinical practice will move on and say, I'm not going to wait for FDA to bless this with an indication. I'm just going to do this because the weight of the evidence is such, and that's how we practice medicine.
Dick, do you have thoughts on, I know it's hard, it's easy to jump into sort of adequate and well-controlled and what the FDA will think about the evidence, but before we do that, just on just the observational sort of component of this in terms of rationale.
Well, so I think we have to remember that RCTs are seen as unbiased for two reasons. First, there's the a priori treatment randomization, which separates the treatment assignment from anything that could be related to outcome. And then there's the post-randomization issues of that are usually corrected by blinding. So again, you want to separate treatment outcomes and measurement of those outcomes from the treatment assignment. Now, we all know, we've all been involved in a lot of trials, that the post-randomization challenges can be pretty big in terms of attrition and unblinding due to side effects and things like that. So we're already often in a world of thinking about corrections for those kinds of issues. And when we think about when we would do when real-world evidence kinds of studies, observational studies, would make sense, it's when we think we're going to be starting to be in that world anyway, and the real-world evidence brings other benefits. And I think we also have to realize that since randomized trials were sort of originally blessed, in 62, if you want to go back there, real-world analysis has come a long way, and our ability to do that has come a long way. And as long as, so to your question, as long as we can do two things with the real-world studies, I think we, one, want to make sure that the hypothesis we're testing is already based on fairly good prior evidence. So clinically, it's a credible approach. As a sponsor, you want to come in with something that's, it's not, the skepticism isn't too great to start with. So there's a plausible clinical mechanism going on there. The other thing, though, in real-world data is we have these behavioral mechanisms, the treatment assignment. And that's where something like clinical equipoise has been mentioned, is hopefully the treatment itself isn't too biased. There are other things we have to think about in the real-world behavior around measurement of outcomes and attrition and other things that could spoil a result, that we can think about the pretty well evolved methods to deal with those things. And so we want to be able to, again, as in any science, have an underlying mechanism for correction that's believable, and then is backed up by all the quality process things that we've talked about. But so those are the two fundamental elements I'd look for in setting up an observational study: credibility of both the clinical thing, so it's a related indication. There's pretty good prior evidence on the clinical medical side. Also, that we can make a strong case that our analytic approach is based on behavioral things we understand.
Yep. Great, thank you. Any other comments from other panelists on this particular question? Peter, go ahead.
Just because I didn't want Mark to leave me in the dust and along with the FDA, so maybe I should comment on that. So I think to a certain extent, well, and maybe just to underline what Richard said, which I think was important, which is the starting point for this was that when the requirement for clinical trials, typically randomized clinical trials, was put in place, it was at an era where the reliability of observational data was exceedingly weak. And obviously there were many disasters that befell our society based upon observations that were not credible but were taken to be so. And that really launched the era of rigorous trial designs, and 314.126 really outlined the criteria that we would use to look at the credibility of trials and whether they met the adequate and well-controlled standard. Fortunately, obviously much has changed. I mean, we've been, over the last actually decades, not just the last years, in an era with improved methodology and better understanding of how we conduct these kinds of real-world evidence-based trials, both observational analysis as well as randomized pragmatic trials with a lot of experience. And I think FDA doesn't want to clearly ignore that type of progress, but we also want to understand where it's gotten us to. Are we there yet? Are we able at this point to apply our understanding of how we can get data from these real-world data databases, the analytic methodologies? Are we ready for prime time with regard to adequate and well-controlled standards? And I think that's the discussion we're going to have throughout the day today. In terms of where one can use real-world evidence, Jacqueline, I think, mentioned already some settings where FDA has looked at this data and used it for regulatory decision-making. We've been doing that in the setting of rare diseases. In oncology, that's making significant progress. Friends of Cancer Research data that Jacqueline mentioned really is moving us forward in that direction. So we're there, and certainly in terms of safety update labeling, we've been there for a number of years. I think it becomes a practical issue around the kinds of data that we can extract. I mean, we have to have the patients with disorders and with endpoints that can be acquired from these databases, which is not always the case, often is not the case. We have to have patients who have the appropriate exposures with clinical equipoise. We can't be comparing patients on one therapy and patients on a completely different therapy where physicians are making markedly different decisions about why they're treating those patients. We have to apply appropriate methodologies. So there's many things that we need to do to get us towards that. Where we can find that kind of data, I think FDA is in the process of really looking at what the criteria should be, what we need to look at to determine whether those kinds of evidence can be sufficiently credible and can meet the adequate and well-controlled standards. So I sort of see this as a work in progress, that we don't yet know where this will go, whether we'll be able to use this evidence more broadly, but I think we're open to these kinds of discussions.
Great, thank you. Mark, do you want to?
So, look, a long time ago, we used to think about it. A drug was not on the market, and then it got the blessing from the FDA, and it was on the market. And everything was fine, but it was not everything is fine. We still had to learn much more about a product. And I think we're also in that same analogous situation now with real-world data. It's not like we're going to reach a bright line where we say, real-world data is good enough now, we can use it for a regulatory context. I think we need to talk about how we evolve going forward, because as someone who I have enormous respect for, who I'm looking at right now, said, you'll know it when you see it. If you have a really good study that shows something that is really credible and believable, even if it's an observational study with a large effect size, you're going to believe it. So how do we get to that place? Now, there has been enormous progress on the methods, and the data has substantially improved, and it's continuing to improve. It's not great, not perfect data. It's getting better. But there are things that I think the FDA can do to set guidance in the conversation with people who produce the data so that we get there faster. So some of these things have to do with asking for the protocol. Some of it has to do with, asking for transparency. I think that we need to raise the level so that we know what good looks like. And that what good looks like has to do with how the study is designed and the process around doing the study. It has to do with the quality of the data that you're using, some of which also includes knowing the provenance of the data, so that, as you're going to hear in the later discussion today, data curation is incredibly important. And we need to be transparent about how data are curated. And for key endpoints, we need to know the provenance of that data so we can go back and review them if it's necessary. So putting those kinds of standards out there is going to ask the data generators and the data analyzers to make a step change and raise their game. And I think that's a good challenge to data generators and data analyzers to make that step challenge. And they'll do that in response to an ongoing conversation with the FDA that says, that's the direction we want to go in because we think that's what good's going to look like.
Just to respond to that, Mark, at many of these panels, I don't always entirely agree with you, but I agree with you. And I think that's exactly the effort we're involved in, which is to really begin to define all of these areas. Now, where that gets us to, I think, is still, we're not completely clear, but we completely agree that all of these areas have to be well-defined, and as we put out the framework and as we put out guidances, that's exactly the intent.
Nancy?
I'd like to raise one question that we kind of glossed over. First of all, I agree with much of what's being said, and it's going to be an interesting day to get to the details. But we talked about when randomized controlled trials are unethical, right? We all just pass right by that because everybody feels they understand that. But I wanted to ask you to think about that for a minute, because I think it's a question, there's the role for observational studies, and we've come a long way from case series and trying to make conjectures from that. So I agree that our methods are developed. But I think we all could recognize something unethical. So you don't want to randomize somebody to a death sentence or the chance of life. That's pretty clear. But I think there's a big category of infeasible or unappealing RCTs that we need to think about. Because if we set a standard that won't be achieved, or as you mentioned, Mark, would take 10 years to get to because nobody wants to participate, that's important. So we see many examples, and I'm sure people in the audience each have their stories of hard knocks to tell, but where randomization isn't acceptable to docs or their patients. So we get these trials where docs won't enroll because they see a clear treatment benefit and they don't want to put their patients in up to a randomization on their future, and patients similarly don't want to participate. So I think that we need to recognize that the infeasible aspect, if it's so unappealing that it quadruples the time that we would get reasonable evidence, that's important too.
Sure. Peter and then Stacy.
I certainly would agree with that, and I think that is a place where using observational data, if it's available, is certainly something worth considering. I just want to remind you that randomized clinical trials that are placebo-controlled aren't the only adequate and well-controlled types of trials that are defined in 314.126. There's a number of other, everything from active comparator controls, which in many situations that are not appropriately ethical or feasible for placebo-controlled trials can be done. Dose comparison trials, non-treatment concurrent control trials, as well as historical control trials, which of course has been used in rare diseases and in oncology where randomization is not feasible. So I just want to point out that observational data isn't the only avenue when we think a placebo-controlled trial might not be feasible or ethical. Not that observational data wouldn't be potentially a solution. I just want to point out that the statute, the regulation actually offers us a number of approaches in those settings.
Stacy?
And I just wanted to follow up on a point that Peter made 4 points ago about, and it's more on the regulatory and clinical side of things, that I don't think we should overlook the fact that the data captured in real-world data sets is oftentimes not the same as the endpoints that would traditionally be required for regulatory applications. And that's a piece that we haven't spent a lot of time talking about within our work groups, but that's an entire set of work in and of itself to try to identify in the practical course of clinical practice, the information that's being captured, what do we find as comparable to those traditionally accepted endpoints, and can we get behind the assessment of those and see them as complementary to those endpoints that we are more familiar with?
Great. Dick?
So I'd just like to follow up. I think we should separate two things. One is when it's infeasible or hard to do clinical trials, and two, when we should believe the real-world evidence in place of those, right? And again, I'd want to make sure we understand both the science and the behavioral aspects of the observational mechanisms that are going on, because that's really the only way, and this is speaking as an economist and the social scientist who has thought about those kinds of things a long time, and we have come a long way in being able to analyze and model those things. They're not perfect, but, nothing's perfect, right? So, we try to do the best job we can. And so if we're going to believe the real-world evidence, even in lieu of RCT evidence, we have to understand the underlying mechanism. I think we've done a pretty good job of parsing it out.
I think we also need to understand, take a hard look at validity, because everything, we're talking about different outcomes and outcomes that matter, but the question is how believable they are. So we started with the concept that real-world evidence used to be pretty bad. And the Friends of Cancer experiment that we did this summer was so interesting. And I love the way Dr. Corrigan-Carre described it as showing that the RCT data were generalizable, and I looked at it as saying the real-world data matched the RCTs. So, but what we see is outcomes, and these were some pretty straightforward outcomes overall: survival, time to treatment progression. But the idea that they're good ways to demonstrate and document that you can trust real-world outcomes.
Great. So we had a lot of good points on sort of when might be an observational study be appropriate. So the potential for harm is low. The proximity to the existing claim is sort of close. Randomization is maybe infeasible, a lot of options to consider there. A big treatment effect when you think that the clinical equipoise is there. And so I guess, now turning to more specifically the adequate and well-controlled standards. We heard already, to get to, adequate and well-controlled with an observational design might look something like, the pre-specified protocol, which is actually spelled out in adequate and well-controlled, having that protocol and analytic plan appropriate and transparent, using rigorous methods to control for the biases that you think are there. And but what are some of the other things that might map closer to adequate and well-controlled for observational designs? And for this, I think I'll go to Nancy, if you've got additional thoughts on that, and then poll the others too.
Well, I think the principles laid out in 21 CFR 314 apply to observational studies and randomized controlled trials. We all need a clear statement of objectives. We need relevant and valid information on exposure, outcomes, and the key confounding factors for the target population of interest. And we need to use data that's been curated to address internal consistency, external validity, and the timing of the information to make sure that we're not missing something because it's stuck being adjudicated. So then we need a design and an analysis plan that makes valid comparisons to provide a quantitative assessment of the drug effect. So what are we missing? And we need transparency. Of course, we have everybody for an RCT or an observational study has to make the method so transparent so that they could be replicated in a different study population or data source so that we can confirm or refute what we saw in the first study. But what's missing from my layman's read of 21 CFR 314 is the randomization. And the question is, how do we get at that? So we've got some pretty fancy analytic techniques, from propensity scores to trying to match people who have the same likelihood of receiving a treatment. We also have tools like quantification of the potential impact of bias. So we can look at an analysis and then say, how wrong could we be? Which is really the question we want to ask. What's the likely effect and how much could it be off? I think that kind of systematic evidence generation will serve us well, just as RCTs have.
Yeah, Mark?
Yeah, so, I think Nancy just put her finger on it. So, an RCT stands on its own. But what does it mean that an RCT can stand on its own? You can say it's an adequate and controlled RCT. It only means that if you pass that magic 0.05 threshold, there's only 5 chances out of 100 that your results came out magically. So is that what we really are talking about? We can talk about whether the p-value is a good thing or a bad thing. I'm not going there. I'm not a statistician. I can't even play one on TV. But the same question then goes to an observational study. So how certain are we of the result of an observational study? And here I think that it's very hard for an observational study, a single observational study, to stand on its own. I think that you want to see replicability. You want to see multiple observational studies done in different populations or in different data sets, all showing you the same answer, and hopefully even using different methodologies to get to that analysis. That's the ideal world. Now, I don't know what short of that is good enough, but because we don't have the same we don't have the same agreement on how much uncertainty is left, and this is where methodologic work needs to be done. We don't have a measure of how much residual confounding is left after we do all of our propensity score matching and whatnot. If we knew, let's say there was a statistical method, I'm making it up, magic, and we say the amount of confounding that's left now is 15%, and most of our observational studies have, good observational studies have a residual of 15%. Then if you had one that was 30%, you'd say, I don't think that's so good enough. Or if you had one that was 5%, you say that's pretty good. We don't have a metric like that. I don't know if a metric like that can be created. But in lieu of creating a metric like that, then I want to have all the other stuff around it, which has to do with the pre-specified protocol, which also means that you register it on a public website so you know you actually did that. I want to see the transparency and we can replicate it. I want to see multiple studies, all of which point in the same direction. I want to see that people tried to use all of the credible methods that we know about to adjust for confounding. And if you do all of that, then it becomes part of the body of evidence. But it's hard for me to talk about a single observational study in the same way I talk about an RCT. But even an RCT, I think, has too much magical properties to it, because even if you pass that magical 0.05, I think we forget the fact that randomization doesn't always work. And sometimes there are different imbalances in terms of patient characteristics because randomization is imperfect. So we have to get away from RCT is great. It may be the gold standard, but it's not perfect in and of itself. And I think we have to get away from, a single observational study, and an observational study is always bad. We have to look at how much uncertainty is left and how much uncertainty we're allowed to tolerate in order to make a decision.
Peter?
So just to point out a few things, just to remind you that the reason we typically require replicate studies is because we do recognize that P less than 0.05 may not be sufficiently robust to really reach the level of certainty that we typically wish to reach. And that's the reason for two studies. Typically, we say best when those studies are slightly different in design so they can inform a bit differently and reinforce each other so that we really can get to a high degree of certainty. When you say, well, we don't really know how much confounding is left, I think you underline one of the concerns that I think we all should have, which is that randomized trials are really set up so that we can draw causal inferences. Now, are they perfect? We all recognize that randomization is a very important tool, but randomized trials can go wrong. There can be missing information. There can be unblinding. There can be all kinds of things that happen. Again, that goes into the reason that we want two confirmatory trials. But in general, when we look at the statistical analysis as well as the overall analysis of the trials, we can get to a high level of certainty. When you say, well, there's maybe 15% residual confounding, the question is then, can we reliably draw a causal inference from observational studies? And that's the challenge I think we face. That's what the whole discussion today is about. How do we get to a level of certainty? Is it exactly the same level of certainty as in two randomized trials where the p-value is 0.001 in both? Perhaps not, but can it get us to a level that meets our substantial evidence standard? And I think as we develop methods to address the issues that you've raised, which is how do we know there isn't residual confounding? How do we know the result is reliable? I agree with you, looking at multiple different databases can be useful, but they can be affected by the same influences and the same confounders that are unrecognized because we can't look for them. And so while replication in observational studies, I think, is reassuring, I think we have to ask ourselves, is it sufficiently reassuring to get us to the level of certainty that we're looking to get to? And I'm not saying that it can't get us there. We're here because we want to find out how we get there. But I think you've put your finger on what the concern is when we look at randomized trials relative to observational data analysis.
Yeah, Dick?
So, completely agree. I think confounding just isn't this total black box usually, though. You have some idea what's going on. If you're talking to a physician or anybody who's out there doing the treatment, you can understand what might be causing confounding. And we have to be sure that we can address those issues in whatever analysis we're doing. And that's why you do it in different data sets, because you start to be able to control for different things and get a sense of what we might be seeing and not seeing in those data. So, and the other fact is, again, it has to be based in a clinically believable story, right? And so if you're generally trying to extrapolate from some RCT results, that you think is probably true, and you want to confirm that it's true in a slightly different population or a related indication or something. So you're not starting from ground zero. And so it's that combination of two things. It's not a perfect science, but I think you realize that. And anyway, so.
So we spent, I think, yeah, please, go ahead.
So Dick made a very important point at the end, which is that at the beginning, I expect regulatory bodies around the world are always going to use observational data as a supplement to randomized controlled clinical trial data. So if you start with a randomized controlled clinical trial data and you know and you believe you have imputed causality in a particular population, and you have more than one of those studies, and now you add other evidence to it, which is consistent with it and makes sense, and you do statistical analysis to say that this all seems to go in the however statisticians do their magic, I think that's going to be good enough. That's what we're aiming to do as we aim to create a learning healthcare system. So if the answer is you can never get there with observational studies, then you're basically saying we can never get to a learning healthcare system where experiential data can be a feedback loop and we can continuously improve the quality and efficiency of care. I reject that. I think we will get there. The question is, how do you do this judiciously? And I think that because you're anchoring it to RCT data, then you don't look at the observational data in isolation.
But I think what we're starting to see, let's come back to regulation before the learning healthcare system. We're starting to see either randomized trials or situations as we move to personalized medicine where it's harder and harder to randomize. And we're starting to see the rise of the comparator, this real world data. So not a single study on its own perhaps, but that middle ground. And this year we saw the approval of a new molecular entity, avelumab for metastatic Merkel cell carcinoma, a great single-arm phase 2 study. And how do you know if that experience is different from placebo when you haven't used placebo? That's when you look at the real-world data. So we're starting to see wonderful models where real-world data is gaining ascendancy, at least for a comparator purpose. And I think that that's a start as we move forward.
And Mark, you're a very controversial kind of guy. So, but I don't think that the FDA or Peter or FDA is saying that you can't get to a learning healthcare system because we have concerns around observational designs. I mean, certainly everybody knows the observational studies and learning from patient experiences in the real-world setting can inform lots of decisions and help learn about the products themselves. And FDA uses observational designs all the time, and safety and other questions. But to get back to, they have a regulatory standard that the learning healthcare system doesn't, like adequate and well-controlled and substantial evidence, and that's where we need to bring these studies to a much higher level for it to support a regulatory decision. One of the things I heard, I think, in the panel, and I don't want to get too much into parsing words, but we will a little bit, is that historically for traditional clinical trials, I like your point about like standing on its own. A randomized controlled trial by itself can be considered adequate and well-controlled. It can stand on its own. But you brought up, even with really good observational studies where you've pre-specified everything, you can replicate everything, you have the best that we know in terms of, balancing comparison groups and reducing as much bias as possible, and maybe even measuring how much bias is remaining, that potentially even then the study may not necessarily be able to stand on its own. And so doing it, like just taking the example of claims data, sort of claims data experiences where these data come from health plans that have various coverage policies and things that sort of direct patients through different avenues of care that might differ from one health plan to the next, that almost always isn't measured, but if you can repeat that study in a number of different policy environments, so one health plan to the other, that somehow, and you see consistency, that maybe that grouping of studies can get to potentially adequate and well-controlled, where that grouping of studies might be able to stand on its own. It almost is like if you think about distributed data systems too, like Sentinel, although used for safety right now, but just the concept of a distributed system that might be used to measure treatment effects, where you are seeing, at one level you have an overall estimate across the range of data partners, but you can also look at what is that variance from data partner to data partner when you replicate the same exact methods and are pulling, almost the same data. Is that grouping, meet the adequate and well-controlled standard? So I do have a question also about, okay, what does it take to get the substantial evidence? But any comments on this so far?
Before you go too far, though, and I'm a big fan of real-world data, but I think you gave the example of looking at health systems, and I think we have to remember that people don't use products just because they can. They use them in systems where a payer has decided to pick up the tab and they've been well reviewed. So the idea that you're going to find the data that you want to, say, broaden your label in observational studies, I think you have to think long and hard about why this product would be used off-label, by who and in what situations, because that in itself can lead to some selection biases that are irremediable. So there's no amount of statistics that's going to change what drives those exceptions. So I think we have to stop back and have a dose of reality that just because we want it to be so and everything could go.
No, Nancy's absolutely right. Look, there are situations where you're not going to be able to solve what you'd like to solve for with observational data. It's just the way it is. Okay, so it doesn't mean that there aren't situations where you can solve for it. And I think what you were reiterating was that, at least my personal opinion is that there's always going to be viewed as a difference, and there should be viewed as a difference between an RCT and an observational study. And that I think that in order for them to get closer to each other in terms of whether you think you can impute causality, I think you need a group of observational studies as opposed to a single RCT. And also, I'd like to see an anchor that it makes sense in relationship to prior RCT data. I mean, I'm not here standing saying that observational data is going to replace everything, but I am here saying that observational data is going to be ubiquitous. It's going to be analyzed democratically by large multivarious groups of both providers, patients, and payers, and the data is going to be used. And so we're going to enter a new Wild West. And I'd like, within that Wild West, to have some guide rails where we say, if you want to see what's really more believable than less believable, these are the characteristics that they should have to make you an educated consumer of that information.
Peter?
Well, I just wanted to clarify a couple of points. First, to say that in general, this space is really where there's an approved drug and where we're looking for an expansion of the label. So we're really not, in general, talking about new approval of a drug. That would be highly unlikely that data would exist or that it would be sufficient. And we're often not talking about a uniquely different indication, although that's possible. We're typically talking about something related to an existing indication, a new outcome within the same disease that's already a granted indication. That said, when we talk about the substantial evidence standard, we're talking about as applied to this granting of a new indication, even if it's a new endpoint for an already granted disease that's treated by the drug. So I just want to be clear that we already have flexibility to say, well, we can approve a new indication based upon a randomized clinical trial or an adequate and well-controlled trial and confirmatory evidence. So there's already the potential to use observational data as confirmatory evidence when it's used in conjunction with a randomized clinical trial or another type of adequate and well-controlled trial. What I think, though, what we're really talking about here is how do we get observational data to stand on its own as a primary support for a new indication, not as confirmatory evidence in conjunction with a randomized clinical trial. That we already have the authority to do, just to be clear. If there is a strong randomized clinical trial with strong statistics, we can take confirmatory evidence, which is not very precisely defined within the statute, and utilize that to make up the substantial evidence standard. So I just want to clarify. I also want to clarify that when we talk about adequate and well-controlled, we're not talking about the totality of evidence. We're talking about as applied to a particular trial. So where we're talking about multiple different data sets, if that's from put together as a single trial data, that would be where we might apply it, not multiple trials to make up an adequate, well-controlled trial, just to be clear.
There's no question the FDA has been using it. You've approved alternate dosing schedules based upon observational data. And it doesn't make sense to do a whole new RCT for an alternate dosing schedule. I think, though, that the number of cases and the frequency with which the FDA applies it will increase over time. And I don't necessarily think it always is going to have to be in the indication. It could be within the ClinPharm section, in which valuable information that is felt to be credible can be added to the ClinPharm section, perhaps related to quality of life measures or whatever. I don't know. All I'm saying is that there is what you can do in theory, and there is what you currently do in practice. And I think you have more wiggle room, and I think that the statutes give you quite a lot of wiggle room to evolve as our understanding of the quality of the data and the reliability of the data and the reliability of the analyses evolve.
Okay, we have just a time check. We do have about 20 minutes left for this session, and I suspect that, I know some of you in the room might have questions and comments, so I do want to leave plenty of time to get to all of those, and I'll ask you to go to those. There's a mic at each end, okay? So start that, but I do want to get to some remaining comments first. So
yeah,
Dick?
I'd like to make one comment, and I think, I'm sorry if I did not infer correctly from what you were trying to say originally about the uses of these data. But I think I would like to respond to one thing, and I think there are plenty of opportunities in real-world data to consider them well-controlled in a fairly good sense. And your example of the payers differentially reimbursing certain treatments in different cases is one of those cases where we look for natural experiments in real-world data, things like proxy randomization, or you have observable things that would instrument the treatment choice, that then you can really start to measure how much they make a difference. Those have become fairly standard approaches in lots of, at least in the social sciences, and I think they have to be carefully looked at more in these cases. They're not perfect either, but they certainly give you information about the treatment decisions and how to control for them.
Any other comments or points by the panelists before I turn to the audience?
I just wanted
Stacy?
to make one more point about this. And we, I think, started to clarify it later in the conversation, but I see this concept of adequate and well-controlled applying to an individual study, whereas ultimately the decision is being made on the totality of evidence and whether the substantial evidence standard has been met. And when you look at any individual traditional RCT, all of these criteria listed in 314.126 are, some of them are black and white, but they're all gradations. And I would suspect that there are lots of traditional RCTs that we aren't as strong on one component as another. And it becomes really important to think about this as a totality of an evidence and not looking at any individual RCT or any individual observational study. I think that, I'm not an expert in the technical science here of RWE research, but my understanding and learning that I have learned from my colleagues is that the the sheer volume of being able to replicate and be directionally consistent with other data that is available is very powerful in itself. So I am not saying that this isn't the right question, but I think that this question about adequate and well-controlled needs to be considered in the context of that broader question of what's the ultimate evidence standard that we're trying to meet.
Great, thank you.
Looking at that in context, we also have to remember that the RCT construct isn't perfect. Otherwise, we wouldn't need two. It would only take one, and they would never disagree. And we know that that's not what happened. So that just underscores what you said, Stacy.
And one could imagine at some point in a magical future, because everybody likes when I talk about the future, that we have everybody in the United States or everybody in the world has all of their data into one data set. And now you do an analysis off that data set. Well, it's going to be hard to replicate that because there's only one country. So you could look at another country, and there's only one world. We could look at other worlds, but you know, there is a logical extension of this. The question is, how representative is the population you're looking at in the data set? What are the biases that are built into them? Can you deal with them? And I think that, again, enormous progress has been made, and I think we are going to find more and more confidence being put into these results by a wide variety of stakeholders.
Okay. Question, go ahead and announce yourself and your organization.
Yeah, Martina Wojtis, I'm with Eljon. I wonder if any of you can comment on the dichotomy that medical errors are a leading cause of death, and on the other hand, we want perfection in drug development and the drug extension process. And it seems like, as a system, those two things seem to be incompatible if society doesn't address that.
Well, okay. So that's a great question, and a lot of part of the discussions that we've been having is, it's one thing to ask a safety question, and we do put a lot of energy into really sort of finding out, is there a safety concern with regard to not necessarily broader medical errors, but severe adverse events associated with drugs, like the Sentinel system that has been developing over time, now looking at sort of additional things on the medical device side like NEST. Safety is looked at in the trials that we've been discussing, and then additional effort in post-market safety surveillance. A lot of energy is put there. For the purpose of this meeting, the big question is whether or not observational studies and real-world evidence that include randomization can bring treatment effect estimates on the effectiveness side, which tends to be harder to measure and to be confident in with regard to these data as safety events are. But I think your question is about broader in terms of the healthcare system as a whole, taking a look at safety issues with the practice of medicine and other medical errors. Is that, great question. Yeah, thank you. Next question.
Hi, Bob Temple. This was all extremely interesting. A couple of things. Peter said that we'd used observational data for rare diseases. Strictly speaking, we used historical controls. It'd be very unusual, maybe there's an example, I don't know, where it's all come from those uses. Now, what the difference is and whether the historical control is more credible, that's another question you can ask, because you're looking very closely at the treated group and not nearly as closely as the historical. However, it's also true that we've used those only where the effects are very large, and that's very important. A lot of people are thinking about observational studies for cardiovascular outcomes, where if you're lucky, the effect is 20%, and there is very little experience with the credibility of those things. I just want to remind people of a paper that Tom Chalmers did in early 80s that compared randomized trials with other designs, historical controls. The differences were vast. My favorite example is that anytime there was a surgical intervention, only healthy people got the intervention in the historical controls. So the people who didn't get treated did way, 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 way worse. I also wanted to ask the group what kinds of endpoints they were thinking might be suitable here. We mostly talk about cardiovascular trials and maybe cancer trials. Are there any other endpoints in real life that we think might be suitable for this? Maybe hospitalization for pulmonary disease, but an awful lot of the drugs we use don't have endpoints like that. So I think that has to be thought of. And finally, one more pitch, which relates to something Mark was talking about. In a randomized trial, you already know what the trial was designed to do. You have a protocol and stuff like that. My worry is that in observational data, someone may have looked around and tried to pick the things that worked out, and you'll never know. How would you ever know whether someone looked at the database and fiddled with it and then decided what study to do? Now, part of his answer may be do it more than once or do it a lot of times, but I think people should be conscious of that and talk about it.
Okay, great question. So the first one was on like endpoints, and Stacy, this was something that you brought up too, in terms of what would be the good endpoints that we think we can get from these observational studies. Nancy?
There are lots of great endpoints we can get. We talked about cancer, we talked about survival, we know that real world data can measure the major MACE, the major adverse coronary events, pretty well. We've seen some good replications with trials. But I think there's the whole field of quality of life and the ability to function, which we can measure extremely well. And that's something important. And I do think, with all due respect, it's a little bit of a low blow to point to badly designed studies and categorize the whole field as bad, because there have been poor designs in randomized trials as well as observational studies.
I'm sorry, you got to read the Chalmers paper. He went through dozens and dozens and dozens of studies in many, many therapeutic areas, and he didn't say they were all bad designs. But I didn't understand your previous comment. How do you measure, I understand how you measure survival and stroke. How exactly do you measure quality of life in real world data? How do you do that?
We ask the patients.
So this is real world data in which you've introduced a new endpoint, and you're going to ask everybody about it. And
Yes,
you
that's
know
an
that they're
yeah,
being asked
yeah.
properly. There's no investigator in these trials. Who's doing it exactly?
Well, I didn't say there wasn't an investigator in the trial, but the art of how you elicit an accurate and believable quality of life endpoint, the FDA has already developed guidelines for patient-reported outcomes and when you have validated scales and the like, but that information is only going to come from patients. And you asked about outcomes we would see in real life. And I think those are the kinds of things that are extremely important, not just disease progression or the change of a surrogate endpoint, but the practical impact on life.
Nobody would dispute the idea that you can randomize and do PROs. Everybody likes that idea. I'm talking about data already collected, observational databases. That's really what people are talking about. I don't see how you're going to get PROs in there.
I think what we're starting to see is that the concept of an observational database maybe started with claims. We've all been working with health insurance claims, and we understand those are for billing, they're really straightforward. But once we've moved into the electronic health records, we're seeing really interesting applications where they're not just straightforward, why did you come to this visit and what can I bill you for? But there are a lot of standard questions that are used in certain applications and clinic settings that do get at these data systematically. So they have systematic collection of things outside the traditional expectation of a chart notation.
Okay. To me, those sound like randomized trials in a real-world setting, and everybody likes that idea fine.
Well, mine didn't have randomization.
Hey, Bob, just to say that quoting from an article from 1982 makes me feel older than I even am. A number of others have looked at the comparison between observational and randomized controlled trial data, including the Cochrane collaboration in 2014, and found that in the vast majority of cases they came out with similar results. And I know that there are two different initiatives going on right now where they're trying to replicate the results of RCT data in observational data. I think this needs to be looked at again. I think Tom Chalmers is one of the greatest people we've ever had around, but I'm not sure I'm going to hang my hat on an article from 1982.
Well, you read it first before you decide. But you're right, we have a very important project going on with Sebastian Schneeweiss, where we're going to be looking at his attempt to replicate results of 30 randomized trials. Maybe doing each one twice, and I'm sure that will be very informative.
And in terms of your second point, you can be sure with an RCT that you have the protocol before. There's a porosity that's happening in RCTs now. A lot of companies are doing in silico simulations of studies and changing their design before they actually conduct an RCT. Not saying that's exactly the same thing. However, that's why we advocate for pre-registration on a public website. Not that that's going to be perfect, but if we create a culture where it's known that it's expected, then only people who know that they're breaking a norm will be doing it. And I think we can get to a much better place if we require pre-registration of observational studies, should they want to be considered for decision-making by regulatory bodies.
I was merely making the point that you can't really know whether they've noodled around with the database and found something.
And, we've
How
also
are you
known
gonna know?
people in RCTs who've diddled around with the database, and we've known
Except I think-
RCTs that have been
but
reported,
Mark,
which have not been fully reporting out all of the results, or not even reporting out the results. So, I mean,
But not when they
I don't
come-
think this is
not
all
when
that
they-
clean.
But doesn't this
But Mark,
also
not when
come-
they come to us.
But doesn't this come back to replication, Bob? And so just say somebody explored in their data before they declared it and came up with a hypothesis. And that's the way science works. You come up with a hypothesis, you leave it for somebody else to confirm or refute it. Once, twice, that's how we learn. So to me, particularly with the advent of artificial intelligence or machine learning, as well as the human-guided learning, we are going to make discoveries, and we want those discoveries. So I'm not as concerned how you came up with a hypothesis. I want to know if it is replicatable.
No, I'm not
Peter,
talking about a
Pete,
hypothesis.
so I wanna give Peter a
I'm
chance
talking
to-
about looking at the trial results before you did it. That's what I'm talking about.
Okay, Peter?
Yeah, I mean, just to underline, I think a concern that Bob's putting on the table that I think is important, and we need to be aware of it, and there needs to be some way that we address it. Because of the number of databases that are available, it is certainly possible for someone to do the same analysis in multiple different databases, see which ones are positive, and then pre-specify the next one. I mean, is that so different from what one might do in small clinical trials that are randomized? Perhaps not dramatically so, but I think the risk is there, and that will be something that has to be considered. So I don't think we could ignore that as a risk for going forward using observational databases. Two just quick comments. I think it's worth underlining, we've been talking about the use of observational data, retrospectively collected data, not prospectively in general, though that also, I think, is an important issue. And I think we should remember that pragmatic trials, trials in real-world settings are a whole other topic that we haven't spent a lot of time on, but really are something that more work needs to be done, because I think the risks there, you mentioned both randomization, Richard, as well as the blinding, both being very important in avoiding biases. I think the whole question around how we do pragmatic trials, if they're non-blinded, they don't have to be non-blinded, is an issue that really also needs to be worked out. And the final comment I'd make is I think one of the things going forward is the potential for enhancing the electronic records of patients. So by incorporating elements that are harder and more appropriate for research type studies in large databases, we can begin to, I think, harvest that even in observational data. We're hardly there. I mean, clearly there are some early efforts at this, but we're hardly able at this point to pull out patient-reported standard endpoints from the databases we have access to. But maybe in the future world, those kinds of questions will be routinely asked, and if they're validated and appropriate, we may be able to utilize that. But again, that's well into the future as opposed to our current status.
Stacy?
And that's the part of the initial question that I wanted to comment on, because I've already mentioned, I think there are a lot of gaps in the data that we currently have available. although I think it's amazing to me the amount of data that are in unstructured fields in electronic health records, either labs or scans or those sorts of things. And I think we have a lot of experience in cancer and cardiovascular outcomes of exploring those. But I think there are others that companies are starting to look at. One example that comes to my mind is diabetes. I mean, we do have an inordinate amount of information in EMRs on blood glucose, on HbA1c, on weight, and those I think are a wealth of information to potentially evaluate those future treatments. I think another opportunity is in the evaluation of rare safety events and the possibility of looking at those in registries. I think a final thing, it kind of gets back to some conversations I was having before the meeting. I think the future of this is going to really rely on some infrastructure changes as well that lead healthcare like systems and healthcare professionals to be collecting information at the point of care that is more conducive to regulatory decision making. A few years ago, I know there were some conversations around Alzheimer's disease and the potential to include measurements around the memory models in some of the Medicare visits. Those sorts of things, I think, are where we need to go in the future. But all of this, I think, has to be precluded with solid conversations between the data owners, the industry, the regulators, the healthcare professionals to identify what are the appropriate leaps to make between the traditionally accepted regulatory endpoints and what's feasible to be captured in real-world practice so that we can then build the infrastructure or identify the places where those data sit.
Sure.
Just comment on the randomized pragmatic trial. So we've been hinting around that, and I'm fascinated by this as a tool and the concept that even in health systems we can randomize and then have the possibility of doing our follow-up, in some cases exclusively through existing records. So it's a pretty exciting time. But we always get caught up in the concept of blinding, and I know we've had some lively discussions on this. But I think it comes back to a little bit where we started about how wrong can we be. So the question is, there I think you have to look carefully at your outcomes. When you have very subjective outcomes, say depression, the likelihood that a reporter, whether it's the physician or the patient, is influenced by the knowledge of treatment so much that they can that will drive a behavior is much more likely with the subjective outcomes. But with things that can be measured objectively, HbA1c and other measurements like that, I think blinding we need to step back and take another look. Now, I know that there are critics who will say that there's evidence to show that the lack of blinding can actually influence HbA1c, but not for long. I mean, it's not, you can't believe that your cancer is going to go away and it'll go away. It shouldn't be that simple. So I think we need to start to be much more pragmatic in our assessments of when you need to blind and what you need to blind.
Yeah, and I think that starts a whole different conversation. I'm not quite as sanguine as you are about whether unblinded trials can be more free of bias. I think it's not just in the hardness of the endpoint, it's in the concurrent interventions, the way the patient behaves, which can clearly influence outcomes, including HbA1c, but perhaps not hard outcomes like cancer. So I think those are the conversations. What are the endpoints that are unlikely to be influenced by concurrent interventions, patient behavioral differences that might occur in an unblinded trial? Moreover, where there is clear equipoise, that is to say, where there are two treatments being compared in an unblinded trial, where there's no preconceived notions in general among the patient or the physician of there being superiority, perhaps there might be greater confidence that differential interventions, co-interventions, behavior, et cetera, would impact the outcome. But I think those are really important conversations to have, because I think you pointed out, Richard, that it's not just the randomization, it's the blinding, both of which are really important in avoiding bias to allow us to make causal inference. And so I think there really needs to be some detailed conversation about what we can get out of unblinded pragmatic trials and what we cannot.
Okay. We're a bit out of time. We'll go to one more question. Go ahead.
Just a quick comment and a question. Panagiotis Mavros from Janssen. So we mentioned before about the need to reproduce real-world evidence in multiple studies. And sometimes we have the impression that we can do that at the push of a button. At the same time, this generates lots of problems because all we're going to do, we're going to generate so much evidence we're going to have a hard time consuming. And coming from different sources, coming in different populations, we actually, the variance in these point estimates may actually cloud any kind of value that we potentially get from this real-world evidence. It appears then that given the inherent problems, both from a methods perspective, the data perspective, the clinical context, we may have to be more deliberate when we do real-world data studies to inform any kind of regulatory decision. And maybe we need more discussion around a fit-for-purpose study than maybe we need that there to save them for a clinical trial, just because all these other factors that we have around. And once we do that, maybe we don't need an excessively large number of replications of our data. And finally, there are newer methods, for example, negative controls, that we can use to assess if the remaining confounding. There is also networks of data which may allow us to reduce the variability in the data that we can utilize to increase our confidence as to whether the data is sufficient to inform the decision we'd like to make. We shouldn't forget these aspects.
Yeah, great, excellent points. It seems like the panel agrees with them too. So, so thank you for not a boring session. This has been really, really helpful. A couple things that came up. One particularly is the role of replicating clinical trial results in observational studies or observational data sources is one that we didn't really talk about, but we are in this afternoon. We have a session, particularly we'll hear about the project that was specified at FDA and the Operand, which is also another project that's doing that, and we'll hear a number of other activities that are happening in this space. So thanks for a great session. Thanks to all of you. We're going to go ahead and take a 15-minute break. So come back at 11:05 promptly for the next session. Thank you. I'll ask our panelists for this next session to come on up and join me on the stage. Your stuff's up here. Want me to mix it all up? Yeah. Okay. Okay, welcome back. We're going to go ahead and turn to our recently released white paper on real-world data quality and relevancy. Hopefully you were able to get a copy from the registration desk. It's the orange one. We'll start this session, and I've got our panelists up here, and I'll introduce you in a brief moment. We are going to kick this session off with a bit of an overview for the central themes in the white paper by Dr. Christina Silcox, a managing associate here with us at the Duke-Margolis Center for Health Policy. Joining me up on this stage, our panelists are Paul Bleicher. It's close. Okay, I saw the nod. It's close enough. Chief Executive Officer at Optum Labs, Courtney Davis, Head of Real-World Data and Analytics at GlaxoSmithKline, Meg Powell, CEO and President of Target Pharma Solutions, and Amy Abernethy, Chief Medical Officer, Chief Scientific Officer, and Senior Vice President Oncology at Flatiron Health. So welcome to our panelists, and go ahead, Christina.
Thank you, Greg. So like Greg said, I'm just going to be going over some highlights of our new white paper, characterizing real-world data quality and relevancy for regulatory purposes. So Greg already went over the revised RWE framework with you, and we're really beginning to be closely looking in on that orange data considerations box. Looking at whether the regulatory real-world data set is fit for regulatory purpose. So I wanted to start by just going over the definitions that we defined for you guys last year, where we defined real-world data as data relating to patient health status and or the delivery of healthcare routinely collected from a variety of sources. So again, this is the framework that we were, that Greg already went over with you. Here we're really going to be focusing on this orange data considerations box, where we're closely looking at, is the real-world data set you'll be using fit for regulatory purpose? And by that we mean, is the data relevant, and is the data of appropriate quality? So I want to quickly just go over some of the common types of real-world data before we start with this next slide. And what type of data you would expect to find in each one of them. And this is something that we actually go over in Appendix A of our white paper, where we list out the common sources of real-world data sources and what type of data. And so none of these will come as a surprise to you. One example, of course, would be electronic health records, which is something that people are very excited about. And what kind of data could you expect to see there? You might see symptom lists, you might see diagnostic test results, you might see patient experience data, prescribed prescriptions, unstructured clinical narratives. You also might see some administrative claims databases. You might see diagnosis and procedure codes, dates of services, pharmacy dispensing data, enrollment windows and when patients were actually enrolled in particular plans. And then we also have a new type of data that we talk about called personal digital health applications. And this stores patient-generated health data, or PGHD. And so what type of data you find in those type of applications really depends on the type of application itself. And so some applications will have patient experience data, some might have personal medical device data, some might have consumer sensor data, some might have things like socioeconomic data, meal tracking, prescription tracking, activity tracking. So it really is going to depend on the type of application in that sense. But this is really a source of emerging real-world data that we think is going to be really important. We also make a nod towards public health databases, and by these we mean less of the type of public health databases which are aggregating claims data or aggregating EHR data. We kind of call that something else, and we're going to get to that later in the presentation. But more public health databases like the National Death Index and the EPA Air Quality Index. And then we also want to mention, of course, that there are an enormous amount of emerging sources of data that could become really critical to real-world data. And those might be things like genomics, proteomics. I think I read in a paper the other day, exposiomics. so all kinds of omics that I think that we're just beginning to understand, but could become really important moving forward. And so in Appendix A, we really list those things out. And then Appendix B, we actually spend some time, particularly for those first 3 sources of data, going through some of the benefits and challenges around selecting and using that type of data and what kind of concerns you might have that you might need to address, either during the selection of the data process or during your methods for cleaning or analyzing that data. So let's pull back a little bit, having defined RWD, let's pull back a little bit to the big picture. What do we mean when we say fit for regulatory purpose? And what are the components of that? And so when we say fit for regulatory purpose, what we mean is within a given clinical and regulatory context, a real-world data set is of sufficient quality as well as being relevant, robust, and representative so that it can be used to produce real-world evidence that can answer the question at hand. And so determining if a real-world data set is fit for regulatory purpose is always a contextual exercise, and I think that we went through this with this idea of the framework, where the assessments of the specific data characteristics contributing to an overall determination of what meaning and degree of confidence can be derived from the resulting real-world evidence. So these characteristics can really be grouped into 2 categories, and we call those data relevancy categories and data quality dimensions. And each of these are really important, equally important to the determination of that a data set is fit for regulatory purpose. And why is this? And why did we separate these out in the way that we did? And it's really for the simple reason that you can have the highest quality, most perfect data in a world in a database. But if the key elements, the key data fields you need to answer that specific question that you have aren't there, then that database is not helpful to you. It's not relevant to you. So these evaluations of data relevancy and quality will be critical to support the claims of causal inference of treatment effect and the internal validity of RWE, particularly for non-randomized studies, as was mentioned in that last session. So let's talk first about the dimensions of data relevancy. Note that an investigator will really be examining both relevancy and quality multiple times as they work with the data. First, during the selection of one or more data sources to use in the study, and then again when they have that final fit-for-purpose data set and they're really doing that final characterization and documenting that final characterization. So what are these dimensions? So the first one is the availability of key variables. Are the critical data fields representing exposures and outcomes, as well as key covariates present in the data, or can they be algorithmically derived from that data? Are the people in the data representative of the population of interest, meaning that they're representative of the patients who are or who will be using the medical product? And are there sufficient people with appropriate follow-up time in the data to demonstrate the expected treatment effect and allow adequate capture of potential safety events? So once you've determined that the RWD you have is relevant, you next need to characterize the data quality. And by that, we mean, to start with, is the data accurate? But is the data accurate? It's a hard question. And so we have some common measures and surrogates of accuracy that have really been in the literature about this, where you're talking about testing the validity of the data elements and any algorithms used to transform the data. You're checking the logical plausibility of the data. For example, is a particular lab result within the limits of biological plausibility? And you're examining the data consistency within the source, as well as the conformance of the data to any internal, external standards that you might be expecting. After the accuracy element, you also want to be looking at how complete are the data. And not just how complete are the data as a number, but what is the extent and the mechanism of missingness? Because that's really important to understanding the potential bias introduced by the missing data and to identify methods that you might need to use to compensate when analyzing your real-world data. And then finally, are the provenance and transformations performed on the data transparent as the data move from point of collection and into various databases? And this is something that we're going to be going into in more detail. So as I mentioned earlier, study investigators are really going to be looking at these dimensions of relevancy and quality multiple times as the data is selected and processed. So the next part of our white paper really walks through this process of taking data from raw RWD and turning it into a fit for regulatory purpose data set. And the reason why we pulled this graph out is we do think it's really important to understand that when you say RWD, you could be talking about real-world data in any one of a number of places, and that really affects what you think of as the quality and the relevancy of the data. Data that's just coming straight out from an EHR is going to look very different from that data that has already gone through that processing to make it fit for regulatory purpose. So this is kind of the most straightforward case, and we put it on here so it'd be nice and big and you could actually read it. And in this case, study investigators would select raw RWD from one or more provider systems or payers or from personal digital health applications and check to make sure that the key variables they need are present in the data source and that the population is representative and robust. And then they would start a potentially iterative cycle of data processing or curation that's really set up for their specific purposes. And so when I talk about cleaning the data, I mean things like doing logic checks, looking for outliers, characterizing the completeness of the data. When we talk about transforming the data, again, they would be doing this to suit their specific needs, so that might include things like normalization, imputation, calculating derived variables, and perhaps some natural language processing of any unstructured data. And then, if needed, linking multiple sources of RWD, either pulling data to increase the sample size or performing patient-level linkages for data enrichment. And once they're done processing the data, then they end with this fit-for-purpose data set that can be analyzed to produce RWE that can be used for regulatory decision-making. So this is perhaps a slightly more likely scenario of what actually will happen and, when you're trying to produce a fit-for-purpose RWD. In this case, in stage 1, data originators, aggregators, research organizations, or others might start this cleaning, transforming, and linking process as they collect data into research-ready databases. And these would be databases that could be used to answer multiple types of questions. In this case, the study sponsor would then select one or more of these databases and select the subset of data that they themselves need to perform additional processing on. They may need to perform additional processing on to produce their specific fit-for-purpose data set. These database owners in stage 1 can really do a lot to increase the credibility and the value of their data by making available the characteristics of the population represented in their database, as well as information on the availability of specific data elements and individual assessments of the quality of these elements. And particularly information on audit processes, standardization, algorithmic transformations, and workflows during original collection would be particularly useful, as well as records regarding the extent of the traceability of that data. Because of this, it might be efficient to establish a process in which these databases become pre-certified for regulatory applications. And the idea here is that there might be some way of pre-certifying that shows that these databases, these research-ready databases, follows appropriate standard operating protocols that are publicly available and considered part of best practices, as well as quality controls. And this type of thing might end up simplifying the reporting and documentation that study investigators would then need to submit to regulators. So talking about documentation, this is really what the last part of our paper goes over. What information do we recommend be documented as the RWD goes from the selection of the data source through processing and becomes a fit for regulatory purpose data set, ready for analysis and to become real-world evidence? So once the data source has been selected, we recommend that there be documentation confirming that it contains the pre-identified data fields from the study design, that it contains a sufficient and representative sample that can be generalized to the population of interest, that they can report the extent of traceability and provenance on the data that before it was acquired by the investigators themselves, and that they have an initial discussion of potential selection and/or information bias that they think might be concerns for this particular data. During the curation process, it is critical that investigators maintain a data management plan documenting pre-specified operating procedures as well as decisions made as the data is being processed. And this includes documentation around the removal of erroneous data, the purpose, the validation, and the performance metrics of transformations made to the data, procedures for adjudicating conflicting data, both within data sources and between them, if data linking, if patient-level linking is used. We also talk about de-identification, Appendix C. And so all of these are sort of examples, and we do go into much more detail in the paper, so I encourage you all to read it. And so finally, once you have your fit-for-purpose data set, then you'll need to do a final assessment of the data set to really to show that you've examined all the issues that are important. And so the most important of that is really the assessment of potential information and selection bias in the data set, which, as discussed in the previous session, is really crucial to selecting the correct analyses when you're turning your RWD into RWE. You should have a discussion of the impact of the assumptions and procedures used while processing the data, an assessment of the accuracy of critical data fields, and an assessment of data completeness. So those are the highlights of what we go through over the paper. And before I conclude, I do want to quickly thank our working group members who were an enormous help to Duke Margolis in crafting this white paper, along with the RWE Collaborative Advisory Group, and also the external experts that attended our workshops on this topic. So thank you all for coming. I'm really interested in hearing the panelist discussion, and I'm going to turn it over to the 5 of you.
Okay, great. Thanks, Christina. Okay, so what do you all think? Maybe I'll just turn and start with Paul in terms of this white paper and all the work that you've contributed and your team has contributed to the discussions that led up to this. What's your major takeaway from this paper?
Well, I think to begin with, I'd like to summarize a little bit of the driving forces, which we heard in the last session, and I think that I had to look down when Mark Berger and Nancy Dreyer were speaking because I thought they might have taken my notes, but in fact they were in my backpack safely tucked away. But you know, to summarize, we currently are in a world where we're recognizing that clinical trials are artificially structured in a sense and inadequate to completely describe the clinical efficacy of medical products. Electronic health record data is ubiquitous. There are legislative and regulatory priorities that are being set by 21st Century Cures Act and PDUFA, et cetera. There is an increasing cost of medicines that we need to address. Time to availability is absolutely essential. We have new tools which we can use. There's increasing personalization of treatments. There are alternative data sources available, and there are many new RXs, and I think Mark pointed this out, biologics and cellular therapies that have complex, unexpected, and adverse event that can't be predicted in the same way they can in clinical trials, as well as a demand from consumers to be involved with and understand and make choices in drugs in a different way than they have before. And I believe that we're going to find that clinical trials are insufficient to meet these needs. This is a very important paper that begins to build on what was done last year and now begins to lay out what data need to look like and how we need to understand that data in order to be able to use it for those purposes. We can't have the mindset, I think, that real-world evidence data is going to have the same type of quality and the same type of control that we get in clinical trial data. They're different. And we are, I think, looking a little bit like, if you think about where clinical trial analysis came from, it came from agricultural field trials originally. And our real-world evidence is more like field biology. Both things create meaningful data, publishable data, important data that explain things. But with field biology, you often go on the abundance of evidence, and Mark pointed this out many times in what we talked about, what he talked about earlier, that it's going to be a group of trials that are going to be important. And so in describing quality, and I want to focus on quality as, you know, in those two boxes here, the complexity of quality assessment is n-dimensional in real-world evidence. It's a little bit like Swiss cheese, if you will. There are, especially longitudinal data, there are holes here and holes there and holes there. We have to come up with a completely new language, I think, and a new way to document the metadata that describe this. This is the beginning. We've actually set now, in this paper, we've set out the places where we need to create this language. But the next task is going to be to develop the language, and it's going to be around provenance, raw data characterization, transformations, representation of accuracy and completeness. And we're at a crossroads. We can actually let that happen in many different places where data are being developed, and then over time we can begin to look together and find some standard, or we can set the standards early on. We can say this is the time at the beginning where we can begin to think about how to describe those things so that a reviewer, whether they're at the FDA or in a journal or some other place, can quickly look down and understand a particular type of data has a particular way of being described. And so I'm very optimistic that this paper actually creates the opportunity for us to take this a lot further. But I think we have a long way to go.
Great, thanks, Paul. Courtney?
Hard to follow, Paul, but as a member of the working group, it is a pleasure to see it printed out, I must say, because I think this, Christina, I think it's version 7, at least, of this document. So it did sort of start nice and tidy, and then it went very big, and then it contracted again, and I think we should be proud that we we got it to where it is, and I do think, to Paul's point, it has laid out some very common sense steps that I think we would all feel comfortable having at our fingertips when we're beginning a project, when we're in the middle of a project, and when we're reporting out a project. And I think especially when we're beginning a project. So we didn't talk much today yet about evidence planning, but I think that this kind of a white paper is a huge boon when you're starting to plan your evidence for a new medicine, because I don't think you're going to wait until you've gotten your first approval to start thinking about, oh, how are we going to get that next indication? I guess we better start planning a real-world evidence program. Actually, we want to be planning real-world evidence programs at the same time that we're planning our clinical trial programs, because ideally, a lot of the same data we can build over time and actually build up those layers of credibility so that they're not just a huge burden to us when we're beginning to generate real-world evidence, that actually some of that same data that you're using in your clinical trial program, you can continue to use. So I think that was one of the big takeaways for me as a member of the working group. I think the focus on transparency from this white paper, again, is really helpful to all of us that are trying to increase the utility of real-world evidence, especially beyond safety, where it's been very impactful to date. So that I think this does help us to lay out, especially the documentation. These little blue boxes, again, we spent a long time trying to edit them down to the most important principles without making it a cookbook. I hope we got the essence of them down there, but again, it's not actually rocket science that you have to document. We all do it as part of our day jobs, but it's just saying what are the most important things that we all know we need to document and document well, and again, mostly pre-specify as much as we can in our documentation.
I think the data linkage part, to me, again, is another part of this white paper that I'm very proud of, because I think increasingly these linked data sets, which are richer, are going to be where we will be learning the most about the relevant real-world evidence, because you have to have the endpoints in there that matter, as well as the data that helps you characterize the indication well. And we know that in many instances, the claims alone are not sufficient for that. So we do require linkage, and as soon as you start trying to link, you will lose patients. And that's where we get more concerned about selection bias, particularly, and/or confounding. So I think that starting to talk about what's needed for a linked data set is also a really important part of this document and the beginnings of the conversation, because there's a lot more to go. And I will pass it over to Amy.
Sure. Amy?
You want me to
Yeah.
go next?
Yeah.
All
There's
right.
a lot of linked data.
A lot of linked data. So hi, Amy Abernathy. First of all, one of the things that came through to me was the importance that technology is going to play in helping us solve many of the requirements outlined going forward. So, in the last panel, we heard about step-level changes in the data sets. I think that's how Mark characterized them. And some of that's got to do with how technology is changing our ability to curate and generate those data sets, as well as link them. But it's also how technology is changing our ability to document the characteristics of the data sets, so that we understand how to best use them. So a couple of thoughts here. One is the language around provenance and traceability has come up several times, but this idea of being able to maintain complete chain of custody and curation for the data, I think, is going to become increasingly critical. If you're doing work with a data set around patients who've got lymphoma, and now the question is whether or not those patients have been adequately allocated to the right designation of type of lymphoma, perhaps need to go back to the actual path reports of the individual patients to document what was in the path report, or maybe even get all the way back to the pathology specimen in the slides may be required, and the ability to follow that through the data set is something that we can do now and should be requirements. I think that we're going to see more and more the role of artificial intelligence, whether that's NLP, ML, other versions of AI in our work. And there's a critical need to make sure that we are using AI judiciously, and we have the same transparency standards in AI as we have in any other aspect of by hand or other data curation. So we need to constantly go back to this idea of using technology and also expecting technology to work for us. We've talked about transparency being key. I think that the ability within the maintenance of the provenance and chain of custody to drill in real time when you need it is something that should be a feature of transparency, as well as the ability to get back to the business rules that were used for transformations and derivations, the understanding of exactly how things are calculated. And I think these are aspects of where technology will play a role. Another aspect of where technology is going to play a role is automating data quality reports. Paul, I think, brought up an incredible important point here about our need to get to standards of how do we consistently document characteristics of the data, but we also should get to standards of how we consistently automate the reports, that we're looking at reports and documentation in a consistent way, again, across data sets. And I think we need to get really granular here, especially when it comes to things like endpoints. Endpoints came up a lot in the last panel. So how do we understand and think about things such as validation? How do we understand versioning, because endpoints and their characteristics are going to change over time? And how do we understand together logical gold standards that we're going to pressure test our benchmarks and endpoints against? My last 2 points are about things that I think are perhaps inadequately represented or missing. One is the importance of recency. The need to get to data sets that represent up to the last second, if not last minutes or last month, is becoming increasingly important. And not only is this about making sure we understand the follow-up available for patients within data sets, but also being able to understand how recent our data, our variables, and how much can we rely on them. And I think this is a key requirement. Standard of care in oncology is changing rapidly, and we see diffusion of innovation curves that are less than 18 months. So we have to understand what's going on right now. The second is that as we start to move towards worlds where retrospective and prospective data are going to be merged, where retrospective or passively collected data are going to be a part of pragmatic clinical trial data sets, we need to acknowledge the fact that the characteristics of the variables change in those different perspectives. And so when we document information about variables, we need to think about it, the characteristics as it goes across time. And my last point is I'm struck by something that came up in the last panel and we're seeing more and more at Flatiron. And that's that in the era of precision medicine, when we thought about real-world data 10 years ago, real-world evidence or clinical experience evidence, we were talking mostly about comparative effectiveness research and large-scale data sets. Mm-hmm. What we're seeing right now is data sets of 20 and 30 people. Often the data sets that are most important are the ones that are characterizing the smallest populations, and the need to go back and understand exactly what patients are included in those data sets. And frankly, complete clinical narratives that are appropriately de-identified is going to be more and more important as we try and understand what all this means. So, thank you.
Great. Meg?
Well, Courtney thought she had a difficult task following Paul. I have the impossible task of following the three of them. So I don't know that I have much to add to the comments that have been made to date. I think first and foremost, I haven't been involved in the working group or the writing group, and so I've been sitting with this paper probably 72 hours. So that's either a huge advantage or disadvantage. But the piece to me that was most striking is that we're still very much talking at the macro level. And so the question to me, especially around data relevancy, is how do you bring these stakeholders together to really drive into the nuances at the individual disease and patient level, and how can we expedite those discussions, or at least parallel those discussions along with kind of these macro guidelines and framework that we're putting in place. So,
great. So I guess one of the things that struck me as we've gone through this whole process of it sort of starting small and getting big, and then I read it and then it got small again, was something that in hindsight is quite obvious. And I purposefully put the slides back on this one because as we see studies that are published with, claims data or electronic health record data, so the traditional real-world evidence studies that get published, when we thought about, okay, it's important to document what gets done to the, well, of course. This stuff is usually in the protocol or the statistical analysis plan, like, if the research, if the endpoint is a derived variable, you describe in the protocol how you're getting that new variable. If you see missing information in your cohort, the protocol describes what you do with. But what we usually don't see described, and typically how this happens, is it's unlikely that the researcher is going to source claims data from providers themselves. Or usually the researcher isn't sourcing directly from an electronic health record. That usually there are organizations and really good people who know how to manage data that create these research ready databases, which is reflected in stage one. So you've got the source data coming in, and some group of people who know how to handle data or organizations do a lot of the sort of curation, cleaning data, and then a researcher might purchase a data set or might work with that organization to actually develop an analytic file for their cohort where they do all of those things that you see in the protocol. But you usually don't see in the protocols what actually happens in that data curate, from the very source to that research-ready database, what happens there? And it was very clear in our work group that stuff is really important, but it's not usually reported. And most researchers, maybe they don't ask the question of what actually happens to the data along the way. And Amy, you've pointed out like the maintaining the chain of custody, or at least making sure that you know exactly what happens to the data in that first stage. So maybe expand a little bit, if any of you want to, on like what should we be worried about, or what kinds of things should we be looking for in that sort of first stage?
So let me start. All right, keep.
Amy?
So I make a lot of data. So, ultimately, issues as it relates to how data sets are generated are fundamental here, right? So not only how data are sourced, so where do they come from, and what are the potential biases that just go into that? So it's critical that you get into that level of detail of where do the data come from, and how might that generate differences in what is understood among the data. I'll give you a very practical example. At Flatiron, we have what I call high-quality sites and low-quality sites. So we have some sites that have very, very well-structured, high-quality data, and other sites where the data is not as consistent. And so being able to have that kind of characterization of the data set so that when you're living in the second bucket and you're doing work with the data set, you can now do a sensitivity analysis to understand the implications of the source of the data. So I think there's this critical need to make sure that we document all the different aspects of the data sets and then make that completely transparent. So at Flatiron, it's a part of a knowledge database that goes with every data set. One thing I do want to sort of suggest maybe we think differently is that it's pretty critical that the people who understand the core characteristics of the data are involved in the research. It worries me when those two things are so disconnected because there's a lot of key features about the data that should go into how you design studies and how you plan the analyses that are going to go into that. And I get concerned when those two things are divorced.
Great. Any others to comment on that?
Yeah, it is very challenging, especially when you're thinking about electronic health record data, because there's a sense, especially for those who are in the clinical trial arena and are used to fairly structured data. And if someone says, "What time did someone get to an emergency room?" that there's a time that's recorded. If you're trying to do time to, intervention or something like that. But the truth is, when you go into an electronic health record and you extract the information, there are often 5 times at one institution and 7 at another and 8 and 2 at another that are the time they walked in the door, the time they spoke to the first person, the time they talked to the triage nurse, the time a physician first saw them. What time is that? So a decision has to be made by someone who's creating a research-ready database, a third party that's creating it and it's going to be usable. That we're going to choose this time, and that decision is made in every time that you get that piece of data from every different institution. So if you've got 80 institutions, there's 80 different decisions. Now you use a standard, which is going to be the time of the speaking to the triage nurse, but multiply that by every one of the of the data elements, and even think of something as simple as glucose. You'd love to know there's fasting glucose. At some places, fasting glucose is labeled. At some places, it isn't. We ran into places where actually animal specimens were run through the human lab. So you had animal glucose, and CSF glucose actually was run on the same machine. And you have to create rules and ways to be able to differentiate that. So the question does come down to how much documentation are you going to require? And at a certain point, you have to think about whether certain vendors of the data or certain aggregators of the data might have to become validated themselves or certified in some way and trusted, if you will, because otherwise, there's a Borges story that I remember studying in my freshman year of college about the person who was unable to forget anything that ever happened and actually couldn't develop new events because they spent all of their time thinking about all the other events in their lives that had happened. Typical Borges kind of story. But I mean, it comes down to that. It does become impossible to document it in the same way. In a clinical trial, there's source data, there's a CRF, and someone goes into the source data and picks it up, and a monitor comes and says, "Where did you get that from?" They point them to it, and they agree with it. That's not possible in the same way with real-world evidence. So,
Paul,
Meg?
I will just, I totally agree with your point. I will say, I think there are iterations of that, though, and you can certainly do that on a subset of the data set, or very specifically talking about the 10 or 12 variables up front and documenting and being very clear on that. So, at Target, we actually do some risk-based monitoring on some key pre-specified efficacy and safety endpoints. And granted, there are going to be a lot that we're not doing that on, but we feel, you talk about high quality, low quality sites. We talk about high quality and low quality components of our data set, based on the monitoring and the investment that we're making that goes into that. Now, we're talking about thousands, not millions of patients, and so it allows that, us to do that at this scale. As you continue to scale, I understand that will be challenging.
That's a nice concept, actually, which is how much confidence do you have in a particular set of data, like the Cochrane criteria or whatever. You might, I think that could be something you could ask a data provider to create, high confidence, based on a number of factors.
And every variable has its own characteristics. So as you think about data sets, you need to be able to say not only this across a table or this across a data set, but this across each individual variable, because variables differ.
So I guess, Paul, you brought up this concept of pre-certifying the database. It's mentioned in the white paper as well. And just sort of background, but I want to get your guys' thoughts on this, is that the idea is, if you look at these two stages, that stage one, the sort of processing that happens by a research organization or a data organization, is typically independent of the exact research study. That then from this research-ready database, then you pull analytic files for your cohort and you make changes to the data as needed per the particular research study that you're doing. But that first stage is almost like the same time, every single time. And there's so much to report out that, would a researcher who's submitting a package of real-world evidence that includes real-world data have to report out on all of these things every single time if half of it is routine and always done? And the idea was, well, if the organization that's producing this research-ready database could somehow get certified, or there'd be some sort of credentialing body that would say, okay, this research database, they have published their SOPs in terms of what they do with their data. It's been shown that this research database can produce analytic data files that are really good ones. That there might be a reason to have this sort of certification process that just might make things easier for that researcher to report out. But wouldn't be required. So nobody was saying that, okay, you cannot derive an analytic file unless you're, for regulatory purposes, unless it came from one of these pre-certified organizations or databases. It was just that if you did that, it would simplify some of the reporting. But there was some, concerns about that among the work group, and may not, some of those things reported out might be proprietary. There might be some concerns about what gets reported. But can you guys give, your thoughts or, some commentary on that part of it?
And also, just to add to that, the burden on a small high-quality organization that doesn't have the financial resources to get certified and to do everything that's required, but might have a database of extreme value and quality.
So,
Anybody?
as a small organization, I will address that. I think there's no question that it would add additional burden, but to be frank, we're putting that burden on ourselves today. And so to have that we might be doing too much. We're consenting all of our patients. As I mentioned, we're doing risk-based monitoring. We're doing everything we can to what I would say to the far right of the extreme of trying to ensure quality. But perhaps we're taking it too far and at the trade-off of less patients, and so, or less data in our data set. So I think to whatever extent we could be more specific in a framework and work towards that, it would help us all speak the same language, which quite frankly I think is the whole purpose of this discussion in this paper, and so we would welcome that opportunity.
And sort of thinking about it from the point of view of Flatiron, but also more broadly, first of all, pre-certification in some ways is similar to how we think of data sets right now, right? We already have in our mind data sets that we understand the properties of. I'll just use SEER Medicare in my own life And so that already then allows us to understand, and these are the things that can be done with that data set. And so being able to now have a certification process that gets us prepared for a series of regulatory use cases, I think is pretty important. I will go back to this kind of core point that versioning is going to be really key, because data sets change very, very quickly. Variables are changing, and we need to be able to understand what versions we're looking at. This also would be helpful because it would help us acknowledge the fact that data sets are continuously growing in several dimensions. They're growing in terms of the longitudinality of any individual record. They're growing the total number of records, and they're growing the total number of linkable data sets. And so a certification process would allow us to then acknowledge that and go to the next step.
So two things I would sort of add to this discussion, playing off a couple of things, I think, Amy, that you mentioned. First of all, if someone is doing a study on the, and I know this is not a regulatory study necessarily, but the process of someone receiving care for an MI in an emergency room, and they really need to understand the data of hitting the administrative person, the triage nurse, the physician, and whatever, they're not going to be happy with a database that just takes one of those times and dates and uses it. So it seems to me that that there's an opportunity to create a structure in which the user of data actually specifies what their key variables are, and how and demonstrates that those that the data they used was fit for purpose for those key variables very specifically for whatever reason. So I think that that might be something, not necessarily all variables, because that becomes almost impossible as a burden, but a well-known certified database might not be sufficient for a particular type of study. The other thing that I really like is the idea of, it's not just versioning, I want to go beyond that. Many years ago when I was at Humedica, about 8 or 9 years ago, and we began to do this work, I fell in love with the concept of reproducible research. Especially the way that Frank Harrell, you can look this up on the internet, he might even be in the room, I don't know, I've never met him, but in any case, it describes it, and going even further, the idea of taking the exact data that are used for a study, taking the description, the metadata about it, the programming that goes into the tables that are used, and the programming that goes into the text using either LaTeX or some of the more modern things which actually use with Microsoft Word. You push a button and the programming works off the data, it produces the tables, it produces the text around it, and creates a published or submittable copy, a submittable report. Having that all then archived together so that somebody can essentially take that out and run it again and have confidence in it is, I think, the best way to do that versioning. And now that data storage is not so expensive, it's not unreasonable to actually consider doing something like that.
But maybe there are some steps to get there, like making sure we know the data are high quality and making sure the observational methods are trustworthy. So we're trying to get, before you push the button, making sure.
Greg, I will just jump in as the one person here who doesn't actually, isn't a data provider. I think I'm in that unique spot. I'm usually in the second part of the diagram, so I'm starting with the research ready data. I think that the discussion about credentialing is a really interesting one. On the one hand, it sounds really appealing because it would give us confidence that there has been this sort of upfront work done, very transparent, it's all documented. We sort of have these agreed on checklists or procedures that we all think are the fundamental ones, and it's just laid out there and we can all see it. But I guess, we have to talk about who is going to be that independent body who actually would do the credentialing, and in some of our discussions before, we got a little bit hung up on that part. So I know we're maybe not
Yeah.
going there at the moment, but I think that is something you have to think.
Greg would like to be there.
Well, so a lot of questions would need to be addressed, like, not just who would do it, but what would we be looking for? Like, what are the what would be the features of something that would be considered, certified? Because you could argue so many times that, well, you certify it for what? Just because it's certified doesn't mean that it can be used to support a regulatory decision. And so we'll be defining what is it certified, and maybe it's just about, like, the methods used for curating the data, follow best practices and have, are appropriate for producing research data, not necessarily for regulatory purposes, and there might be other things, and I think we're going to talk later on this afternoon, like, well, if you've demonstrated that you could reproduce clinical trial results with that research-ready database, does that give you additional confidence or not? And should that be part of it? And questions like that. So it's just an idea, one worth debating. Any other comments? Well, I think we might have, not yet, but we are going to open it up in the next minute or two to questions from the audience.
I think my only last comment is with regards to, we talked about the provenance of the data, and obviously the paper mentions that quite a bit. And so just because data today has not been validated does not mean you can't go back and validate it in the future. So if in very specific data endpoints that are quote unquote low quality but become of a more higher importance as you continue to do that work, I think, you're getting at that with versioning is too. If you have a good source document or the ability to do that, you can, data can move up, if you will, in that definition. So,
okay.
There is one elephant in the room that we haven't discussed that we spent a lot of time talking about that I'm just going to bring up, which is the FDA's expectation with clinical trial data is that it's possible to go back to the original patient. And especially in retrospective observational studies, we're talking about de-identified data. And while I think for a significant safety issue, I know that the FDA has the authority to be able to break that, I assume. Bob, you can tell me whether that's in fact correct. But for just general purposes, I think the process of de-identification may make that challenging, if not impossible, to be able to just go back to the original patient data that was used and de-identified to create a database.
So there are technical solutions now that can help solve that and be able to, in what I would call a HIPAA clean room, be able to now get all the way back to the individual patient level. In fact, when we talk about these small cohorts that we're working with right now in the precision medicine space at Flatiron, we're going all the way back to the individual patient level data and those narratives. It's critical that you work within the confines of HIPAA and the confines of patient safety, privacy.
Okay.
But yeah.
We'll go to we have more people than last time. Okay, so we'll go to a question over here. Go ahead and state your name and organization.
Hi, I'm Stella Chang from Allscripts. So having been affiliated with a number of data providers, the one thing that keeps me up at night is the engagement and consistency of the original data contributor, whether that's a payer or a provider. Because even if the data contribution is done in a very passive manner, there's usually several avenues that a data contributor can opt out. So I'd love to hear the panel speak to some of those challenges and any recommendations.
I'll start with this one because this was both a focus of our research at Duke and then something that we focus on at Flatiron. Two observations. Number one is if you try and force curation up front within the clinical setting, there's only so much curation that can happen before the quality actually falls off, because you can imagine that you start pushing buttons just to get to the next screen in the electronic health record or other places. And what we learned is that it's approximately 5 variables in any clinical visit you can structure. So you really have to come up with processes that work around the clinical activity but don't interfere with it. The second is that every single variable has different characteristics about when it was collected, and you actually need to incorporate that in your understanding and documentation. A good example is performance status in oncology, which is very, very reliable on the first visit and at times of treatment change, but it has problems with cut and paste errors at every single time in between. And so you need to be able to involve that within your analyses. And any data provider should be able to give you that level of detail for every variable.
Okay. We'll go with Mark.
Hi, Mark Berger. So if we could have full transparency around the curation process and have ability to have provenance in it, that would be a huge step forward. But the idea of certifying a database is like the idea of validating a questionnaire. A questionnaire is never valid. You just have a lot more experience in using it. I remember back in the old days when there were some data sets which are today considered very, very good data sets. When we started using them back in the early 90s, we found a lot of inconsistencies in them. And we'd go back to the data aggregator, and they'd say, oh, I see we have to fix the data set this way. And no matter how much curation you're going to do, there are still anomalies that are going to be found by researchers who get their hands dirty up to their elbows and start playing with the data. So, I mean, I appreciate the idea of certification, but I think the first step is no one right now, as far as I know of the data aggregator, actually reports out on a continuing basis a report about their curation methods and provide some descriptive statistics about missingness, blah, 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 that would make it just a general way of benchmarking one data set versus another.
So, Mark, we can roll the tape later on because I'm sure this is being recorded, but I think actually what I said was not, I know that was said, but I think what I said was certifying the provider of the data and their practices and their quality, which would involve, of course, somebody looking at doing an audit of the data and understanding how they do it, and the provider certifying that that's the type of quality and approach that they take. I think that's probably fits more with what you are thinking and is more appropriate, I think.
I agree with that comment completely. So if I misspoke, I certainly at the provider level, not at the data set level. I think just even some standard SOPs, I mean, GCP light, if you will, of moving forward would be helpful.
We'll go to Gracie.
Hi, Grace Eberman from Genentech. So 2 comments on this. I know I was one of the ones in the working group of sort of a proponent, and I don't know, do we call it pre-certified or sort of annual audit of the data? I don't know what's the right word, but it almost seems to me like 2 comments, right? The small providers. I think if you're providing the data once or twice a year used for regulatory submission, there's probably like, why do it? I agree. We just do this once or twice and a year. But if you're a company that you're going to know your data is going to be used more than 3 or 4 times a year, then the same efforts that would be repeated, how do we streamline them? And one way would be to sort of sit down and think about it. What would be the process from the FDA or other regulators to sort of look at that and see, is there a way to streamline that process and do it on a biannual basis or something? So it's hard to say, is it pre-certified, because it's not forever, like the versioning. There has to be a process that continues. So thank you.
Okay, great. Over here.
Yeah, Cyrus Mehta from Cytel Corporation. I'd like to point out one of the dangers that can exist when the producer of the data is transferring it to the actual researcher. It's an important principle when you are selecting controls from a database in a non-randomized study, that especially because you'll be using techniques like propensity scores later on for making that selection, that you not have access to the actual efficacy variable. If you have access to that efficacy variable, then you can bias the study. And it's really up to the researcher to have a good SOP to document that he didn't have that type of access. And that's why I was a little concerned with a comment of one of the panelists who said it would be a good idea for the researcher to work closely with the producer of the data, or maybe I misunderstood that comment, but I don't think that would be a good idea. I think they should be kept separate, and there should be a way of documenting that no access was available to the actual primary endpoint efficacy. But it's a cardinal principle that's emphasized again and again in our statistical meetings.
I 100% agree. And you can wall that off, so you can actually sort of wall it off in the same way you wall off confidential information within companies. So I think that's a different issue, but you're absolutely 100% right.
Yeah, and I think that at least some of the discussion on this was, or at least feeling behind it, was that for the producer of the data, that might in real-world data sources, we're talking about the hospital system that is using the electronic health record, or the research organization that might be part of a payer, where they understand a lot of the things that happen to the data along the way, or like what we were talking about on the previous session, the coverage policies that might bias the data in some sense. And so having an understanding of that is important for the researcher, and those two groups talking to each other in terms of, so that the researcher knows all of these things that may not be apparent in the data, that's an important thing. But I think that there might be a couple other things that we're referring to when we think about the producer of the data being closely related to the researcher. Yeah.
So Cyrus, I love that point actually, because I'm a believer actually that a lot of retrospective observational real-world studies should be done in a blinded fashion, with the efficacy variable blinded. I am told by people who routinely do these studies that that's quite challenging. I'm also told that that is far from the standard. In fact, it's extremely unusual, uncommon, if not unlikely to occur. But I think we should think about things that are done for a regulatory purpose, that we should think about blinding as another very strong piece of armor for the quality of the results.
Okay, over here.
Paul Copeland, Johnson & Johnson. Continuing on that line of the efficacy variable, it seems for having research-ready databases, one of the things we need is validation of the efficacy variable. It seems that that's something that FDA almost always requires, and that's a huge lift. And also bringing in what Amy talked about, predictive analytics and machine learning, we can increasingly improve our efficacy variables through machine learning and predictive analytics to use combinations of codes or medication usage and ICD-9, ICD-10 procedures to build a stronger efficacy variable and then validate that. One project that is ongoing currently is to validate opiate overdose as a measure that can be used in different databases. And 13 companies are collaborating to do that and sharing the costs equally, so that the cost and the effort is distributed. And whether you would consider adding that as development and validation of key efficacy variables with things like MACE, stroke, or opiate overdose that are important and used a lot, as a way to perhaps consider collaboration across different public-private partnerships to foster more research-ready databases. I know IMEDS is doing some work in that area, and perhaps there could be other venues to do that.
Can I add one comment there? I've often thought of this concept of validation assays, so that we have consistent ways of validating variables across data sets, and we're running that assay also across time so that you can follow the performance of variables across time. And it behooves us to publish those validation approaches or those assays and make sure that we all understand them, agree them. With one very practical example is the National Death Index as a way of validating mortality data in different data sets, and we just need a series of validation assays.
Over here.
Thanks. My name is John Birch, and I'm a member of the Mid-America Angel Investment Group in Kansas City. I have two questions that both relate really to the underlying economics of what we're talking about here, which I think is getting kind of scary. First is the definition of real-world data still includes the term routinely collected from a variety of sources. And it's the routine aspect of that that I think is coming under question. We're talking about a set of processes here that are less and less and less routine, certainly in the delivery of healthcare. And so some kind of extra effort is going to be required. The Salford study was full of that. They all have emphasized the cost of their labor, how labor intensive it was, the cost of the nurses who had to do all the work. And I know we've heard from other sources, Amy, that the labor intensiveness of what Flatiron did came as something of a surprise as well. I guess you've survived, though. But anyway, that's one question, and I think that it suggests that not just a technical solution is going to be sufficient, but a socio-technical one. New jobs of some sort. I'll just leave that out. The other is the reliance on algorithms for doing a lot of the data curation, or just the data analysis, and the need to make those transparent. We're beginning to overlap here with something that I hear a lot of from the device side of the house, the SAMD project, the software as a medical device. Where really it's the same problem. You can't be doing the kind of algorithmic curation that we're talking about. I mean, what you are doing then is you're using a software as a medical device to help you process the data, and data is used for medical decision making, so therefore it's a device. And so I think there's a lot of additional costs that are coming out here that are going to really, really change the economics of this kind of research. And just one last point that I'll end with is that there is only one real world out there. And Mark made the point earlier, what if we all had our data and it was longitudinal? You know, there are so many uses of that data. What we're talking about in this room right here, the regulatory uses, is a small window of the needs for that data. And I think when you look at all the possible uses, the economics actually are quite favorable, but we have to deal with the kinds of problems at the source that you all are talking about here. Yeah. I'll leave it at that.
Yeah, great points. Amy?
There's two extra costs. One is that there's an increasing demand for clinical understanding in these data sets that we really haven't not had to have when we were talking about predominantly claims data sets or other aspects. So the need to actually bring in that new jobs of some sort, the new clinician who's a part of that has become progressively more important. And the second is, as we think about software as a medical device, it's actually software plus people that become the medical device. And so I do think that there's a whole new list of jobs out there that we just don't understand yet.
Great. Next question over here.
Yeah, hi. I'm John Lashinger, CDRH FDA. We have a different model that really shows the value of an underlying infrastructure. And basically, in cardiovascular devices, we're blessed by a series of 9 different patient registries that capture 95% or above of all the patients that receive cardiovascular devices in the United States. And so it's a very robust all-comers database with no patient selection. And we can use that to make regulatory decisions, and we have, both for label expansion and for new device approvals. Specifically where other trials couldn't be done. We know from experience that to enroll certain trials takes years and years and years to get just a few hundred patients. And using the registry, we were able to get data on 4,000 patients in a period of 6 months from real-world use. So from those kinds of things, we find it very useful. It's an infrastructure we didn't pay for, but there's also infrastructures in every other, in Sweden, Japan, UK, France, and Canada that are now being formed based on these models, and we just had a meeting last week to try to even link all these up so we can do international studies as well. But the underlying infrastructure is very important and allows you to do a lot of different things once it's up and running.
So the comment I'd make, first of all, as a recovering immunologist from way back, I'd say that registries are the F1 hybrid of clinical trials and electronic health records. The interesting thing about registries is that you can get structured information and data about things that you really can't get out of the electronic health record, and you can't, and you'd have to have a ridiculous clinical trial that probably wouldn't work to get that kind of information. And so I think that where there are registries, they're incredibly valuable. Combining them with patient experience, patient-reported outcomes, information obtained from, someone was asking earlier about what are some of the outcomes you could use for things that don't have hard A1C-like outcomes. Time to return to work or something like that is an achievable outcome that's effective, it's clinically meaningful, et cetera. And those are the kind of things that might be combinable with registry information, and some of these concepts still apply.
Yeah, and in fact, we have a couple registries now that are adding patient-recorded outcomes like KCCQ and things like that to their databases and can collect about 85%. It's not 100%, but it's still pretty good. And the other thing we are doing now also is linking these databases. The good thing about cardiovascular devices is you either die, you have a reoperation or reintervention over the total product life cycle. So by linking these databases, we can follow patients for 10 or 20 years now. So
I'm really glad that you made that point. So our business is effectively registries. We don't use that word because registries mean so many different things to so many different people, and so we don't like to use it because each one of you probably have a slightly different definition of what that means. But I think that our belief is that it's a very important segue into the next generation, right? I don't think we're going to just flip a switch, as we heard in the earlier panel, and that this is going to be nirvana. And I think what registries do is allow you to syndicate or get people together to support and put together the infrastructure that not one person could do or not one stakeholder could do on their own, give you higher sense of quality in the data validity, allow you to do some things like consent so that you can link to other data sets in a consented manner, collect PROs directly from the patient, et cetera, and be that kind of stepping stone, if you will, to validate these larger, more comprehensive data sets, for the future and help us feel more confident moving that direction.
Great. Okay, we'll take one last question.
Thank you. Albert Kraus, I'm with Pfizer. Question actually relates to the slide that's up there, and it's much more technical, but it's around interactivity of stage 1 and stage 2, given the different groups involved. And it's probably coming from my experience in more rare diseases, oncology, and segmented, targeted therapies in small subgroups, which I think Amy mentioned. We in certain cases are narrowing down to small groups of patients because of the nature of the rareness of disease, and there may be great ways to study things that we just haven't been and can't in phase 3 randomized trials of a traditional sort. But the question really is the comment made about the need to drill back and get all sorts of information about disease, patient, prior treatment, performance, and other things. So I'm wondering about interactivity between stage 1 and stage 2. Have you written it in comments about where we need to grow, improve, and do in that?
I personally believe the interactivity between stages 1 and 2 is fundamental. It's very hard to be an investigator, so I'll put my investigator hat on and be an investigator and plan studies and not know the details of the data set. It's also very hard to be on the data set provider side and try and get all of the things right that you need to hand over a priori. And so that conversation back and forth, we need more of this, or we're going to need the following additional details, I think is pretty critical. That being said, one of the key points before about appropriately deciding where you're going to divide labor so that you are not cherry picking, that you are not exposed inappropriately to the endpoint, et cetera, is going to be also very critical.
I mean, I think that usually the phase 2, a series of feasibility analyses is done to get to go back into phase 1 to even decide which data sets you're going to use or which populations or health systems, are feasible. So, I mean, I don't think you can have a complete protocol for phase 2 without going back many times often to the first part.
It's interesting, though. Our academic collaborators all think that it can be phase 2 independent. That's often the mindset. And what we find is as we start working together, it becomes 1 + 2.
Okay, so that wraps us up for this session. Thanks for sticking with us. Thanks to the panelists for a great conversation that we've had. We're going to go ahead and break for lunch. It'll be we'll come back exactly at 1:15 for the next session. Like I said this morning, there are some restaurants around, and we have a map outside. So thanks to the panel, and have a great lunch. Okay, welcome back, everybody. For this next session, we're going to hear from a number of organizations that are also doing a lot to improve the science and policy of developing and using real-world evidence. We're going to do things a little bit differently. I'm going to ask the presenters to come up one at a time, and then at the end, ask them all to take a seat here to field any questions from the audience. But kicking us off will be Jeff Allen, President and CEO of Friends of Cancer Research. Jeff?
Great. Thank you very much, and thanks for the invite to join the meeting today. Obviously an important topic, and I've heard a lot of progress has been made. So I'm really excited to be here and share some top-line results from a pilot project that we had the pleasure of working on with several of you here in the audience today over the earlier course of this year. But I'll just go ahead and dive right in so that hopefully we don't spend too much time talking and can get to discussion. I know I have a few slides that are really small, so I'll try and just sort of hit the main points here. But essentially, as there's been a lot of discussion around the role of real-world evidence and how it can be used by different parties, what we wanted to do by putting together an initial pilot project was to sort of take stock into where we were in the field of oncology. So we had the good fortune of being able to work with 6 different data partners that are listed at the very bottom of the slide here, who helped us aggregate some information and see essentially how 6 different data sets answered a similar question. And what we focused on was the utilization of PD-1 inhibitors in non-small cell lung cancer. The reason we chose that particular area as our case study was because there are multiple approved drugs in that space. We didn't want this necessarily to be a re-adjudication about the effect of the drug per se, as well as a large population to be able to have robust data sets to perform some of these initial analyses, and also the ongoing interest in terms of what the long-term effect of IO therapy truly is, despite its original transformative potential. Originally, when we set out to do this, again, our goal was not to look at reevaluating the clinical trials per se, as well as it also was not to necessarily showcase the capabilities of our different data partners, but to really try and arrive at an analysis based on the existing data of what could be done in a relatively short period of time. So to the credit of our collaborators, we launched this study toward the end of 2017, designed it over the first few months of 2018. They performed their internal analyses over the spring and shared this back to for public review this summer. So it was a very fast project that I hope provides some information about several different endpoints that can be extracted from both claims or EHR-based data, potentially for the use of regulatory use, but also looking at, could these also be indicators of long-term benefit? With the idea of, in oncology anyway, we're seeing a great frequency to ensure that unmet medical needs are addressed, that initial approvals rely on intermediate endpoints with the need for follow-up, which can be difficult at times. I won't go into a great deal of detail here, but this lists several of the parameters that the data partners help design ways in which to extract this information from their data sets, as well as ways to censor their data to try and begin to compare as much as we could apples to apples around things that could be extracted from their data sets, like overall survival, time to next treatment, time to treatment discontinuation, or in the case of those data partners that were building data off of electronic health records, things like time to progression or progression-free survival based on real-world information. And this is essentially a glimpse at what was found. So what this is intended to show is the different demographics that each data set was able to break down their information. So things like age, histology, gender, and the line of therapy. And what we saw is you can see that the data sets varied in size from a couple hundred to several thousand, but the distribution of the population and the demographics of the population contained in each were relatively consistent, which I think is an important point looking across different data sets in terms of reproducibility of information. It also shows generally that this level of information can be extracted from these data sets with consistency between all of them. So this shows some of the initial results here. Sorry, I'm realizing like maybe my eyesight, just how great it's becoming. It's kind of hard to see, but what this essentially shows between each of them are some of those endpoints that I mentioned earlier that each data partner was able to extract. So in terms of real-world overall survival, we saw essentially about 10 to 12 months on average. Time to treatment discontinuation around 3 to 4 months on average between each of the data sets, as well as the time to next treatment averages within the final column. You know, one interesting thing that became very clear off of this, of course, there's a high desire to look at the typical gold standard clinical endpoints like survival, but it became very clear that this is, while a highly desirable endpoint, a difficult data point to actually get the data for. So I think this was a good exercise, not only for raising the need for better records or better sources of data in order to determine date of death, but also the opportunity to look at other metrics and see how well they correlate to those measures as well. In this, it shows again a couple of the breakdowns based on demographics that we saw here, depicting time to next treatment as well as real-world OS. And at least for several of the data sources, if you compare sort of all the blue lines to all the blue lines and all the red lines to all the red lines, we see some degree of consistency, particularly in things of around age. For example, those 75 and older throughout the different data sets behaved quite similarly, which could be an interesting sort of data point or hypothesis to look at moving forward. This table goes back to depicting what the correlation between some of the real-world or extractable endpoints is towards some of the clinical endpoints, including overall survival here. So the first column, or the one in the middle, depicts each data set's correlation between real-world overall survival and time to next treatment, and on the right, the correlation between overall survival and time to treatment discontinuation. The bottom are the data sets that also had access to this information based on electronic health records and were able to correlate real-world progression as well as time to progression as that correlates to OS. And one of the things that I thought was very interesting in this pilot and perhaps warrants a future study moving forward is the role of time to treatment discontinuation. In our pilot project here, for most of the data partners, it showed a high level of correlation with overall survival, around the 0.8 range. And this actually correlates as well as progression-free survival did as extracted from the electronic health records. And obviously that's an endpoint that has been relied upon for a variety of uses from the clinical setting. One of the last things I did, before I pass on the mic, I thought this was an interesting exercise that we did following completion of these original analyses. So again, each of these bars depicts each one of the data partners' data, and the red lines are essentially the range of overall survival as determined through the clinical trials that were used to approve the different PD-1 agents that were used in this pilot. So what this shows is that in this particular instance, each of the data sets falls pretty close within what was observed in the original clinical trials. And we really didn't know what we would find here. I think when we started this project, at least for us internally, as we thought about the outcomes that we might observe from this, it wasn't. We thought this would be a good exercise in understanding what the real-world treatment effect would be, with the likelihood that it would be diminished as the population being treated became more diverse, had comorbid conditions or other factors that are particularly frequent in the case of the marketed use of an oncology product. But in this case, what we saw is that for the most part, it fell within the range of what was observed in the original clinical trials. And similarly, this graph depicts, again, the different data partners' evaluation of time to treatment discontinuation. The red line in this instance is from a study that was conducted by the FDA in collaboration with Mass General Hospital, where they took the pivotal trials, again used for the approval for these PD-1 agents, and did an internal assessment of the time to treatment discontinuation for those at a patient level for those particular trials. So again, while we haven't done any of the statistical analyses, from a trending perspective, you saw relative similarity between the time to treatment discontinuation in the real-world population, as well as what was observed for the pivotal trials for this particular indication. So just a couple of top-line conclusions from this pilot project was that there is a high level of shared characteristics between the different data partners, independent of the size of the data set. And we were able to break that down to several different parameters that could be useful in the future to think about how different subset analysis could be rigorously performed. The pilot project also demonstrated that several extractable endpoints from the EHRs or the claims data were able to correlate with overall survival actually quite well in this instance. This also showed that the subset breakdown were able to be reproduced relatively accurately between the different subsets, so things like age or treatment effect when separated by gender were relatively consistent by the different data sets that were included in this particular project. And finally, that it was similar to what was observed in the original clinical trials as in terms of the magnitude of the treatment effect between the real-world data set and the clinical trial data set. So I know this is hard to read, but I wanted to include this just as sort of an example of the various next steps which could be considered. I think this pilot project was number one, a great example of collaboration and a willingness of different data partners to come together and answer a similar question, and as well as a great hypothesis generating tool in terms of things that could be followed up on. So we begin to try and think through what would be next to build on this. Do we need to think about reproducing this in a different treatment setting? For some of the findings, artifacts of lung cancer, or some of the findings, artifacts of IO therapy, which, as we've talked to the clinical community, could be true for things like treatment past progression that could contribute to a difference of correlation, say in this particular setting versus general chemotherapy. As well as several methodological considerations that I'm sure will be discussed even later today about things that would need to be further fleshed out to build on this. But I really do want to acknowledge the contributions that have been made, both from helping analyze the data set, but designing this project with experts from CODA, Flatiron Health, IQVIA, Kaiser Permanente, and the Cancer Research Network, a partnership between the Mayo Clinic and the Optum Labs, as well as the University of Iowa and PCORI to facilitate this work moving forward. But thank you very much.
Oh, stay close, Jeff. We're gonna. I think since each presentation is somewhat different from each other, if anybody has questions for Jeff about this study, I think why don't you stay and probe a few of those. We do have microphones on each side, but if you can speak up, maybe.
Yeah, so we didn't present that. Some of the data sets, I'm sorry, the question was whether we broke down the analysis based on PD-1 expression. And some of the data sets have the ability to do that better than others, as well as sort of the probably the mix of the population inherently anyway. So in non-small cell lung cancer, nivolumab is not used in a selected way to the same extent that Keytruda is. So basically what we asked for was really the treatment effect based on the population that utilized those products.
Okay,
Any others?
I do have a question for you. So Jeff, you saw in your results that you showed, some variability among, across the different data partners, and presumably those, the data sets or the data partners that you're using had variations in the makeup of the patient population, as well as, potential differences in collecting data. When you saw, inconsistency across the data partners and also inconsistency with what clinical trials have reported, did you learn anything about sort of why there was inconsistency? Or not everybody matched the clinical, but why? So what, expand on that a little bit?
Well, I think, some of this was an iterative process, and maybe even a process of human behavior as well. We tried very much to lay out as strict of definitions as we could in terms of how to collect the data. And each data set, I think to your point, each data set is different. Extracting the information from each data set is different too, and how to do that. So several of the partners, for example, that utilize claims data, they were willing to share codes and things like that to try and help sort of the partnership and be able to extract similar information from similar populations. But even the analysis, this was something that, as sort of we went through, was an iterative process of, thinking through, did everyone indeed calculate this the exact same way? So even down to that level was important. And I think that's very telling for the future of the studies or the use of this data, is that necessity to have that level of transparency. And at least for someone who is not an expert in the data science to the extent of all of our partners were, I think that was very telling as we think about future policy. What exactly needs to happen in order to ensure that level of transparency from a methodological standpoint, or what can be done in order to standardize different methods, definitions, ways of extracting this information so that different data sets can be more readily compared to one another. Otherwise, you're right, it turns out they can be quite different for very small reasons.
Great.
Thank you.
Okay, thanks, Jeff. Next will be David Martin, Associate Director for Real World Evidence and Analytics in the Office of Medical Policy at CDER FDA. David, if we can go. There you go.
Hi, thanks for including us in this discussion. So the focus of this talk is going to be on real-world evidence demonstration projects which have been initiated in the Office of Medical Policy in response to 21st century cures. But I just wanted to emphasize right from the beginning that there's close collaboration across all of CDER, also including CBER and including the Oncology Center of Excellence in both policy and ultimate guidance development, as well as a number of other demonstration projects which, for sake of brevity, aren't necessarily included in this presentation. So in the interest of time, this has been well covered in other discussions, but obviously it's been said multiple times that there's, in a sense, an existing current benchmark for assessing evidence, and that benchmark was unchanged by the 21st Century Cures Act. And so there's a bunch of tools or sort of routine practices that have grown up, which you can see listed on the slide. And so as we move into an environment where we're assessing the potential utility of real-world evidence, there's a natural inclination to relate those new tools and new procedures back to that paradigm. So that being said, what are the real motivators for our demonstration projects? Well, I think there's sort of three major motivators. One is we want to expand the quality, quantity, and diversity of real-world data that are available. And so you might call that like an infrastructure building type goal. And we've heard plenty about this, but I have a slide coming up. Number one is that you can't get all your endpoints from claims data, for instance, and we've heard many people say that. And so the idea is, we obviously took a look at endpoints that are out there and kind of came to the conclusion that a lot of people have said here that there's going to be probably an all of the above solution, claims, EHR, mobile technologies, other things, wearables. So that's an issue. Another thing is that the more diverse and sort of larger quantities of data you get, you often measure more and more confounders that were previously unmeasured, thus potentially increasing the capabilities of your sort of analysis as well, even in a non-interventional space. So the next aspect of this is there's been a little bit less talk this morning about pragmatic trials and registries, but obviously they've been mentioned. And in CDER's definition of real-world data, those are both included. So we really felt that we needed recent experience, recent practical experience with real-world randomized designs and with registries. And so that has motivated some of our demonstration projects. And we think this will inform our guidance development. And then finally, which we obviously talked a lot about this morning, there's an urgent need to assess the performance of non-interventional designs. And we want to pressure test widely accepted designs. We also want to think about potentially new paradigms that are out there, both in terms of design or analytics, but also new paradigms in terms of regulatory review that may differ from standard practice with the extensive use of real-world evidence on the safety side. So this is just a reminder. This is sort of giving you a sense of the percentage of trial endpoints that kind of come from different domains. And I think you can sort of appreciate from this that there's sort of a smaller percentage that you might tend to find in like claims alone. If you bring in other kinds of technology where you're communicating directly with the patient, you may capture a bunch of CROs, PROs, and there may be things with increasing levels of curation of the EHR where you can really get much further across this whole landscape. So that's just a reminder again of another motivating factor. So now I'll just start going through some projects and I'll try to highlight key points. So first, you've probably heard for several years about the IMPACT AFib trial. And I think, just so you know, this actually is an OMP project that predates Cures. But really, so first, actually, let me step back and say, people want to see progress. So from a progress standpoint, I would like to say that the IMPACT AFib trial has actually sent the outreach letters, and since many of you are veterans from many of these meetings, I don't really need to describe the trial. You could probably describe it back to me. But intervention letters went to approximately 40,000 patients, and this was done by 5 data partners, all working together to make this trial work. So I think this is just a great example of even pre-Cures, data partners trying to work together to really give you, in a sense, that sort of big data capability that's been talked about for a long time. And so this is obviously a very important accomplishment. It leads me to one other thing, which I didn't put this on a slide, but I just wanted to mention that one of our key sort of test beds for demonstration projects is what we call the FDA Catalyst system. And for some people may not have heard of that. So when we use what we call Sentinel infrastructure from the Sentinel system, so that would be things like the data partners or Sentinel Analytics, but then we add primary data collection or primary contact with patients or providers, those projects fall under OMP's FDA Catalyst program. So this is an example of an FDA Catalyst program, again, predating Cures, but still relevant. This is another one, and I need to actually personally thank, which I always do, the Patient-Centered Outcomes Research Trust Fund, who funded this. So this is the FDA My Studies mobile app. This is a means to actually go out and get data from patients. And it includes both the app, but more importantly, a storage environment. And the combination of the app and the storage environment allows you to do distributed studies, whether to work with multi-site clinical trials or multi-database studies. And the issue here is that this is FSMA and 21 CFR Part 11 compliant, so it meets the most rigorous use case for inclusion in clinical trials, and I'll have more to say about that later. So now I want to talk a little bit more about some of the new projects that we're just launching. And again, these are supposed to be highly relevant to the 21st Century Cures use case, obviously of looking at information that might someday be the type of information, or at least the type of questions that would lead to supplements, whether it's SNDAs or SBLAs. So first, when we started thinking about this landscape, we really wanted to leverage other federal investments, and we received a lot of help, both from our collaborators through the Sentinel system, but also through our collaborators at PCORI. And we had some prior work with PCORI in the past aimed at broader uses of Sentinel, and the two groups sort of aligning together. And so Joe Selby in particular, but others on his staff, really helped me and other people at OMP kind of go through the suite of PCORI-funded trials and look for some examples of trials or studies that might relate to the 21st Century Cures use case. So I'm just going to take you through a few of those right now. So the first is the Reliance trial. And this trial actually still has some benchmarks to meet before it receives its sort of final go, no-go decision from PCORI, but right now it's in the process of working to meet those benchmarks. And it's a situation where there's equipoise between treatment with azithromycin, which is guideline suggested for COPD exacerbations, and roflumilast, which is also FDA approved for those exacerbations. And it's set up as obviously a randomized trial. And so FDA Catalyst is going to come in, and obviously we're going to be working with the study team, connected to and learn about multiple aspects of the trial. But we're going to, through the help of Catalyst from the Sentinel Operations Center, bring CMS claims data to bear to help capture some of those primary endpoints that might have somehow been lost if they only had what they currently have, which is direct outreach to the patients in a more traditional trial-like way, as well as EHR data. So here's another one, Limit JIA. So this is a randomized real-world trial in patients with limited juvenile idiopathic arthritis. And in this case, there is sort of a concern in the pediatric literature about, when do you start sort of stepped-up therapy with biologics, and when do you sort of stay with usual care? And so the investigators particularly commented on how they felt that in this particular case, a non-interventional design would not work. But in this case, there are children who will be randomized to receive abatacept, and others will receive plus usual care, and others will receive usual care. And so, again, we want to be part of this, we want to learn from this, and we also want to bring something to the table. So in this case, we're bringing the FDA Catalyst mobile app to the table, and this is going to be used to help capture the hard endpoint of uveitis, which it turns out, is something critical where the ophthalmologists who see the patients to diagnose uveitis are not actually trial investigators, and we need to have a special link to potentially pull that information into the trial. So there's also some other potential work with the subjects are actually being recruited from academic centers that also participate in a registry called the CARA registry, and we may also support the registry. So again, helping us learn from registry experience as well. And then another one of these is Spark IBD. So this is a cohort within a sort of large-scale research exchange platform, maybe more commonly known as a registry, which is actually being used to actually support a PCORI-funded prospective observational study with PRO outcomes. And so again, we're going to sort of fold in with the My Studies app and help them capture these PROs and help feed this PCORI study. And I think that this has led already, even in these planning stages, to some really productive exchanges of information between FDA and the individuals trying to make these studies work. Because I think it's really critical. It's very easy to say, I want to do, or it's very easy to say, I want you to do a pragmatic trial, or I want you to create a registry. It's very hard to do it yourself or be part of doing it yourself. And we think this will be very good for informing, guidance development and policy, and really just helping to keep us as informed as possible. Another issue, another example I wanted to mention is we've aggressively, obviously, been working with OND to really think about endpoints that are potentially more or less amenable to real-world data use. And actually, we had a great proposal, which kind of developed collectively through conversations with the Division of Pulmonary Allergy and Rheumatology Products, DPARP, to really look at, in particular, COPD and asthma endpoints as potentially being ripe for capture in real-world data. And so we're launching a new collaboration. We are going to be looking at a bunch of these endpoints in some Sentinel data through the help of the Sentinel Operations Center. And one of the fun things we're going to do is we're going to try to pre-replicate the Reliance trial, which I mentioned to you as one of those first projects, so that hopefully we come out with findings before, well, I would say we will probably come out with findings before the trial does, and we'll see how those line up. So that brings me to my last, not quite my last point. Second to last point, just want to mention, there's been a lot of talk about data quality. Ignore most of this slide because it looks a little different than the Duke paper, but I had some very favorable impressions from the Duke paper. But I just want to point out one thing. This thing here, the use of electronic records and electronic signatures in clinical investigations under 21 CFR Part 11, this is something to think about. So, because in the Duke framework, when they were talking about data provenance, it's always been important for FDA, and it remains important for FDA to ensure, the authenticity, integrity of the data. That's not going to go away in the real-world data environment. And if you want to get sort of an early window into maybe FDA's thinking, I would suggest you look at this guidance because it gives you some sense of where that's leading. Another thing I'd like to pick up on is I did hear one of the panelists earlier say, because people hear me say that and they're like, oh no, more regulations is horrible, but there are technical solutions to these things. So I want to turn this on its head and say, we worked on building the mobile app. We've deployed it. We already used, I didn't mention this, we actually used it with Kaiser in a test study already. But, when we got done and we built this thing, we were 21 CFR Part 11 compliant, just through our technological solutions. So why would we water that down, and say, oh, well, for real-world data, we don't need, if it's not a trial, we're going to do something lower. We've already built it. Just use it. It's compliant, So that's at least a positive thing. All right, so now we'll get to the final part because, we got to get back to the controversial topic from this morning. So assessment of non-interventional designs. Obviously, the CEDAR definition of real-world evidence contemplates analyses that either include randomized treatment assignment or are non-interventional. They're within the definition, they're within the scope for our evaluation. So actually, you heard Bob Temple mention this, and actually, very quickly, I want to mention both Bob Temple and Mark Levinson, who have not stood up yet, but they, maybe you're going to ask me a question, but anyway, please don't. So they've really been instrumental in working with me to set up a high throughput clinical trials replication project. And we want to basically, right now we've selected 40 clinical trials. We're hoping that after you go through the process of assessing feasibility, that hopefully about 30 will remain, we'll see. And we want to actually do real-world data, non-interventional designs against those trials and see if there's agreement. So the point I'll make about this is we've been working on this for a while. We've sort of canvassed O&D and received a lot of useful input from various divisions. We worked to a priori set up agreement metrics, and we worked a priori to set up a process for doing these attempted replications. And I did want to mention offhand, because we get some criticism for this, we're using claims data as our data source. Right now we have 3 different claims data sources. That was chiefly because this is the first wave. We want to do this fast, high throughput, and basically with some low-hanging fruit in terms of questions. We actually have a follow-on project which will include some EHR data elements and broaden this in the future. So the goal, the timeline, we hope to have 30-ish completed by March 2020. But we are going to be putting information out as this goes, so it's not like, you won't hear anything from us and I'll just pop up in 2020 with, here's your answer. And we're actually starting with sort of the diabetes therapeutic area. So I just want to, this is my last slide, so sorry, we're almost done. But I thought this is kind of worth mentioning because, and there's a lot of overlap here between what you see on this slide and our process. and what ISPI and ISPOR came together and discussed. So I think, Mark, you would approve. So, but essentially, I think, and again, this isn't to make policy, this is demonstration project, but I think you can pick up on some themes here. So in general, non-interventional designs don't work great in the world of a supplement when you sort of drive by White Oak, open the sliding door of your van and just dump it out. Here's your report. What do you think? Can I get a supplement approved? So what are we modeling? What behavior are we modeling in this replication project? And I'll sort of go through these. And I guess I used to think I had good eyesight, but I need some glasses, so I will have to turn away from you. So first of all, there's prospective engagement with FDA during protocol development. Okay. And also during the feasibility and power calculations. And then actually yours truly will have to sign off on the protocol at that point. And then we're going to have final, so there's sort of a feasibility phase. We're going to make an initial triage, so some of those 40 may just drop off. And then there's sort of an additional more significant review when we're coming to agreement with, final definitions related to all these key things that, on the trial side, you're used to people coming to agreements about endpoints with FDA. So this isn't dramatically different. And all of this is, which I heard someone else talking about in the morning panel, is blind to differential outcome. Now, it does turn out we are allowing aggregate outcome to sort of be used for some power calculations and things like that, but no differential outcome. So now I have, the next part is all in red, because you can sort of think of that as then you cross a line of departure, basically. So, and ISPI and ISPOR talked about, it's fine to do exploration, hypothesis generation, et cetera, but when you do, Mark, how do you pronounce an HETE study? Do you guys call it HEAT? Okay. When you get to an actual evaluative study intended for the purposes we're talking about, for drawing inferences about the performance of the medical products, then there needs to be these important sort of lines of demarcation. So then you kind of cross the fabled line of departure, and we're going to first register what we're doing on clinicaltrials.gov so you guys can see that we're about to do it. And then we're actually going to do the analysis and calculate the effect measures, and then we're going to document the findings, and then we're not going to, at that point, decide what our agreement metrics were, because we already, and this is a little different because this is for our special research project, but we already decided what those metrics were, so now we just have to apply them, and then we will apply them. And then the last part I'll mention about this is that throughout this whole process, there's actually an FDA, an audit trail that's visible to FDA. And in fact, in this case, FDA also uses the same analytics suite and has access to the data. So that means that we're going to have essentially a pretend review team. We're like a research review team, for lack of a better term, or a sub-team that will look at each one of these replications and think about if there are any other questions we might have wanted to answer, sensitivity analyses we might have wanted to do. And so that's really my last point, which is, and Bob Temple got up this morning and said, he's concerned about mulligans. Well, I can't remember what language he used, Bob, but just people trying 1,000 times, batting it once out of the park, and then handing that to us. So, and when we heard responses from the panel, and I understand everybody kind of understands that audit trails keep honest people honest. So you need audit trails, but I think a paradigm you'll probably see out of FDA in the future is the old arms control paradigm of trust but verify. And I think you will likely see a need for regulatory agencies to trust but verify through their own replication efforts. And so I just want to give you some of these themes and hopefully give you a window into our demonstration projects. Thank you very much.
We're going to get questions, I assure you. Thanks, David. Any questions for David on his presentation?
Spectacular. So
Okay,
let's start with that.
Okay, thanks. Now I feel better about whatever's coming my way.
No, it's not. What's coming your way is a discussion we've had on a number of occasions.
Right.
Clinicaltrials.gov was not designed for observational data.
Right.
And Deb Zarin has not been the most enthusiastic person I've ever met in
Yeah.
thinking about clinicaltrials.gov as a repository for observational studies. And there are specific issues with registering observational studies and what data you want to, or what information you want to collect in advance of doing the study, and how you, whether you date and timestamp the pulling down the data from a data set or whatever.
Right.
I do think there is a real need. There are as we talked about in our task force reports, there are a number of sites out there where you can register observational studies. It's my personal opinion that none of these sites are ideal. I do think it would be worthwhile to get a collaboration going, saying, assuming at some point in the future we're going to be asking for registration of studies, let's talk about what the data elements should be on a registration site. I don't care who owns it, as long as it's a public site and everybody can get access to it. But there's work that needs to be done. And I don't know what the correct answer is. I don't know anybody knows what the correct answer is. But I think by getting different stakeholders together, I think we can craft something so that in a couple of years, when this starts to become standard practice, we have a home that's really good for observational studies.
Yeah, I concur that there's more to be done there. So thanks for the suggestion.
Hi, David. Nicole Gatto from Pfizer. Great presentation. Can you comment on, you had mentioned that some of those 40 replications that you're trying to do, after feasibility, you will probably, they'll be ruled out, or they won't make it through the go, no-go decision. Can you talk a little bit about what those feasibility criteria are? What are you looking at?
Yeah, so I mean, in like very simple terms, number one, we could have like a statistical power issue, a feeling that, we just really shouldn't basically be asking the question due to sort of the amount of data that we have. The other thing, and this is really where like the practical experience comes in, because people talk about there's no hard criteria in these areas, but I mentioned that we want to sort of pressure test like commonly used methods. So in general, we're going to be using new user, parallel cohort design, new users in both cohorts basically that are being compared, and there's going to be propensity score based methods. And so one of those feasibility things obviously is looking at the propensity score distributions in the two groups, and getting back to what was discussed this morning, if there's a feeling that there really is extreme channeling going on, one of the things that gives regulators comfort with propensity score based methods is that you can essentially get a mathematical feeling that that is actually happening, and you can decide to abort a study, that looks like there's just insufficient overlap and those kinds of issues. So I would say those are probably the two key feasibility issues. There could be others that crop up.
Okay, thank you.
Sure.
Okay, thanks, David. Great presentation. So now we'll turn to Cartier Esham, is Executive Vice President of Emerging Companies at the Bioindustry Technology Organization, or BIO.
All right, and thank you for a correct pronunciation of my name, but everybody calls me Carter, so we can, I wasn't able to pull it off quite well. I don't have slides today. I'm really just here to share with you some thinking that we as an industry have been doing for the past over a year and a half since passage of 21st Century Cures. And I'd first like to state that let's provide a little context, or remind everybody about a little context from the industry perspective of why are we doing all of this thinking about real-world data and how we can use it as evidence. And I think it's important to understand that how we're looking at this is this really has the power to help provide us with information about how patients are treated and how they respond to treatment in clinical practice and when they take their medicines in the real world. Performance of therapeutic innovation is a function of age, comorbidities, real-world medication taking behaviors, or comparison to other treatments is going to be extraordinarily valuable the better we get at this. This will also help us expand learnings when information from random clinical trials may be limited or unethical. So to that point, starting at that context, when we look at when we started talking about real-world data, we realized this is a very large topic. And so what we decided to do is really focus on a key area about how this could be utilized, how real-world data could be utilized in the context of expanding a label. And so what I'm going to run through is some of our thinking on that and perhaps some challenges and perhaps some path forward to help us be able to do that in the future with better efficiency and effectiveness. So we all know that in order to use real-world data to support label expansion, like everything else, it has to rely on the data being of sufficient quality, depth, and relevance so that the FDA can be assured of the validity of the study and conclusions. When we started with that principle, we started to think through what are the factors that need to be incorporated in determining the quality of real-world data. And that includes ensuring that data set generation needs to use established procedures and outcome measures, including validated ascertainment algorithms that will help us identify and or minimize bias in parameter measurement. We also need to ensure that data management and control processes are in place to ensure fidelity of data following transfer and storage, which has been talked about today. And I will note, as I looked through our paper and then was quickly over the weekend reading the Duke paper, the good news is I'm not cheating. We just are happening to say a lot of the same things. So that is a good sign. But again, back to accurate data validity and quality. We need to make sure that data accuracy is assessed by evaluating consistency between independent measures of data values known to measure the same attribute. We also must ensure that sponsors assess potential effects of residual confounding due to missing, unmeasured, or unavailable covariant data. We also need to make sure that the data is representative of the target population that is of interest within an application. Study estimates and conclusions need to include an assessment of potential biases that could generate a false or alternative result. So further thinking along these lines about how we can do better and utilize real-world data for label expansion, we believe that observational or interventional real-world studies are appropriate in many instances to support label expansion. However, there are several factors that can help us determine whether observational or interventional real-world studies can support reliable inferences from study results. And these include biological plausibility of the study hypothesis, the degree to which natural history of a disease is understood, acceptance of the therapeutic or prognostic biomarkers being measured, the clinical endpoints that are observed and captured in real-world data, the amount, quality, and relevance of real-world data or collected during routine clinical care, and the observed treatment effect, and to make sure that the observed treatment effect is supported by a causal disease framework. And these are just a few of the key factors. When we next thought about what would a best practice look like, we really looked at what industry is presently doing and looking at the accumulation of guidance that FDA has provided in many other areas, relevant areas. And they do not, I'm happy to say, include anything about vans and dropping off large packets of data. We do not support that and understand that could be a big problem. So best practices, we recognize that the hypothesis generation does need to occur at the outset of the study design. And use of pre-specified statistical analysis plans that address the specific study, hypothesis, and objectives and explain the details of the primary and any secondary analysis is critical. We also, a best practice is to make sure that consideration and incorporation of several different sensitivity analyses when appropriate to test potential drivers of study outcomes, for example, differences in baseline characteristics, treatment changes midstream or missing data are also important. Transparency in methods and analysis conducted to enable others to better reproduce studies is also going to be very important. This will increase confidence in study findings by testing the robustness of results under different study designs. So when we look at taking all of that into context, it's sort of now what? And we do need to realize that while this has been happening in many cases, while this information has been accepted by FDA in many instances, we are still forging a path forward that we want to be broader, more easily understood, and more easily accepted and utilized, and understood by the patient community and understood by the provider community. So when we started thinking about what are the challenges in that context, we first need to make sure that everybody understands, again, that we're not talking about lowering standards. We need to figure out how we do this in a way that meets the standards, the gold standards that everybody understands today. We also think it's going to be important that, such as this meeting is reflected, that stakeholders continue to engage and work together to figure out how we together can not only augment expertise within the FDA, but also external to the FDA so that we are all familiar with data and methods that we can utilize to generate real-world evidence for the purpose of effectiveness evaluation. And again, I'm saying all of these comments for now in the context of label expansion, but I think these principles go across many utilizations of real-world evidence and data. We also think it will be important that we understand when real-world evidence generated from secondary data sources are identified and deemed to be qualified, so that we understand when it might be appropriate and fit for purpose for evaluating effectiveness in support of label expansions. Additionally, circumstances in which publicly funded data and data from national data resource networks such as CORNET and Sentinel, when they could be available in the public domain, needs to be described and understood. We need to improve data quality through longitudinal integration across data sets supported by robust data curation and structured data collection. And finally, the development of regulatory use cases demonstrating how real-world evidence can provide appropriate evidence for regulatory decisions related to label expansion, including the approval of a new indication or patient population for certain clinical questions and contexts, will be critical for advances to be made. And in this case, what BIO has been forming, developing, and discussing with stakeholders is how we might be able to use, under the pilot programs available under the PDUFA 6 agreement, a pilot program akin to what we're seeing in the innovative clinical trial space, including the aspects of shared learning could be applied in these circumstances to look at label expansion, which again has been talked about. FDA is clearly already doing some great work in this space, but this would enable and unleash the power of industry and their collaborators come forward with ideas and proposals and ensure that work can be done that will help us all better understand how to approach this in a way that uses quality data to answer key questions and enable expansion of the label. So I'm happy to answer any questions today or as a follow-up, but that is what the industry is working on presently. Thank you.
Okay, thanks, Carter. Any questions for Carter? No? Okay, thank you. So our last speaker for this session is Barbara Beer. She's the faculty director at the Multi-Regional Clinical Trials Center at Brigham and Women's Hospital and Harvard, or MRCT Center, and professor of medicine at Harvard Medical School.
So I do want to say that the work I'm about to describe is an actual honest collaboration with Optum Labs, and Bill Crown is going to come up and do part of this with me. So you can get ready to come up, Bill. There we go. So I'm going to describe a project that we've been working on for some time, which integrates a lot of what we've been hearing today and actually follows on from the work that Sebastian's doing at the Brigham and Women's Hospital, replicating 30 trials that David spoke about. We started this work probably, I think, a year and a half or two ago now, and we call it OPERAND, Observational Patient Evidence for Regulatory Approval and Understanding Disease. So we've really sort of tried to think through in a prospective way a lot of the issues of using observational data for much of what we've talked about throughout the day here, and I'm not going to really repeat that. Nor will I see what you mean. These slides are very small when they're up there. And glasses won't help. So I'm not going to repeat the 21st Century Cures Act, but needless to say, we started this back before any work was really done, and hope to contribute to the outcome. So as you can see here, there are a whole host of participants in this project. It is from the outset a real collaboration amongst many pharmaceutical sponsors, and as well as the FDA. David's been an active participant, and we appreciate all his efforts there. The School of Public Health at Harvard is for the National Pharmaceutical Council, Pharma, and Duke Margolis has been there as well. So we really have fashioned this project initially by pulling everybody together to discuss it, to discuss the vision, to work out what we would do. And from the very outset, it's been a collaboration. And then we, after deciding what the approach would be, we then sought funding, and I'm happy to say that it is now funded and we will proceed with the work. So the good news, I guess, for all of you who are tired is this is a data-free zone. You're not going to see any data because we're just beginning the actual part of this work. So the idea is that, that we would take the Optum Labs data and choose a number of clinical trials that for which there is RCT published, then use the inclusion-exclusion criteria from those trials, lock down the data, as it were, in the data from the real-world data from Optum Labs in order to see whether through that aperture of the inclusion-exclusion criteria we could replicate the studies from the RCTs. Now then, that's part 2A. Part 1 was planning all of this. Part 2A is sort of figuring out whether or not this can be replicated, and then gradually opening that aperture and seeing what happens to the effect size, and learning a lot about how we use empirical data in that regard. So the objectives are to figure out how they replicate RCTs, understand data quality and the limits of the data quality in a controlled environment so that we really do begin to understand what this offers, what this doesn't offer, and do that all sort of transparently before we move forward. We're starting with trials that have claims data and EHR data. The claims data, at least one of the two trials, will replicate the same data that Sebastian is using so that we can compare what two different groups completely delinked, and we're careful not to talk to each other about the process and how we're going about it, completely delinked really do replicate or not. And if not, then we have a lot to learn about what we did and didn't do and figure out why and how to proceed from there. So after the claims data, combining that with EHR data to see how that may refine the endpoint and what additional data and challenges RWE does to that analysis itself. Look at the sensitivities and variabilities. We are going to define certain statistical approaches, but then let the teams that do the actual analysis have liberty to apply what additional methodology that they wish to, and then sort of think about how that informs on-label effectiveness versus efficacy. So this is all on-label in phase 2A. It will be beyond it will be on-label, but beyond the inclusion-exclusion criteria for phase 2B. So the idea is now, having defined the way to go about that, we're getting, we put together an RFA. We're sending it to the Optum Labs data partners who are quite familiar with the Optum Lab data, and then choosing 2 independent laboratories that will do the work that we've defined with the trials that we've defined before we've looked at the data. They will register their approaches in ct.gov, absent mark any other clinical trial database at the moment. We do think it's important, like when we think about this, which we've done a fair amount of, we think it's important to figure out how to make sure we know enough about the data that we can register, but not so much that we know the answer and we start, sort of hunting. And the easiest way for us to do that is not to release the data set until it is registered. Now that's easy for us to do because we have separation between church and state. The data analytic arm is quite different than the data holder arm. That's not true everywhere. So in that regard, we think replication is very important. So 2 teams doing 2 trials each, and they will have claims and claims plus EHR. And then, as I said, we're using a variety of methods, which we'll go to in the next slide, and have a common clinical question, a common protocol. They'll have the protocol, they'll have the data, and then they can analyze it. And when they finish this sort of narrow lens, we'll come back together with the technical expert panel, which we call the TEP, to analyze that and see where to go beyond. So the methodology is, as we've discussed here, we're specifying a whole set of statistical analyses, but we know that academic partners will indeed move forward to do their own analysis. What we want them to do is to tell us what analysis they're doing and what assumptions they're making about the data, so that we can figure out, with 2 separate teams looking at the same data, how differently do they approach it. So this goes quite beyond what we've talked about today in terms of data quality and data source, to think about the methodology and the variation of the methodology, which I know Duke Margolis is also working on and we'll come back to a little later in our work together. So with that, we're looking at, a number of the RCTs and choosing studies based on the characteristics that you see here, so that we can ensure that there is sufficient data in the database or the data, set that we have, that there are enough patients that have been treated with drugs for a long enough time that we can do an analysis without it being too restricted or too predefined, and yet not so long that it's dramatically different from the RCT that was published. So it gives us a sort of window. All of these things, when Amy spoke today about recency of data, we're not talking about minutes, we're talking about years, because we need enough of the population to be treated, but we completely agree that this is an issue. And then endpoints that are rather well captured both in claims and EHR. And you can see the other components there. And of course, given the data that Optum Labs has, it has to be US-centric. And I should say that this has been a real collaboration, not only of the people that you saw on the second slide, but also we've been working with the group at Harvard or the Brigham in order to make sure that we share that data advisedly, because we don't want to influence each other before the results are known. We've been working here with the Duke Margolis Center, and again, we couldn't have done this work without the help of FDA. So, anything to add? No, perfect. Well, it wasn't perfect, but good enough.
Thanks, Barbara. So just maybe a question. We are running quite a bit long. I'm going to go ahead and do away with bringing everybody back up to have a panel discussion. We'll go right into the next session. But before we do that, any questions for Barbara or the Operan project? Yes, please.
David Thompson, Syneos Health. Thanks very much. Very good presentations. This is for you, Barbara, and probably for you, David, as well. The whole notion of the replication of trials with real-world data sources, I have to say, I don't want to be skeptical, but I do have a couple of concerns that perhaps you could address. Number one, we know that there's the efficacy-effectiveness gap. So we know and we appreciate in the real world that drugs perform differently. They're taken differently, prescribed differently, patients behave differently in the real world versus trials. And so if we appreciate that and we expect it, then what are we expecting to find when we're trying to replicate the trials? And so, for example, certainly you might say, oh, we want to make sure that the treatment effects are preserved at least qualitatively. But one could imagine a situation where one drug is far less tolerable than the other, and therefore compliance is very bad. In trials, compliance being enforced, it would have a very favorable profile, but in the real world, patients receiving it could do very poorly, and therefore the treatment effects could actually switch. So it leads me to wonder what we're going to learn. And if you were to then say, okay, well, we're going to restrict our attention to patients who were perfectly compliant, then all of a sudden we're not doing real world research anymore. And so I kind of wonder where we're ending up with it all, if you see what I'm saying.
So, David, do you want to answer, and then I'll start? You go, whichever. Okay. We're running out of time. So let me give you my take, or a brief take, which is I think what we'd like to see is directionally that the treatment effect is similar. I think that what we need to learn is how far apart they are, and therefore why. Because I don't think what we want to do is sort of start a path of saying, look, we're going to use this real world data and extend, indications or administration changes or whatever without knowing what issues there are in looking at this. So this is a very much to try and get empirical data to answer exactly the kind of questions that you ask and to know what the limits are of that data. So.
Sure. Okay, so try to be quick. So, yeah, I mean, you raise good points. It's certainly not a perfect approach. I think what I would say, first of all, obviously, is that this is supposed to So, and number one, we don't know how this will turn out. So, this could all turn out that we should never use non-interventional data design. Excuse me. So, but let's just pretend that actually it appears to be worthwhile in, say, one or more therapeutic areas in one of these, one of these replication, activities. I think this is about trying to close that chasm between people who sort of just want to say, well, there's always some unmeasured confounder somewhere, and so you can't ever use this. And then the sort of cheerleaders of this, and as Barbara said, we're trying to get some sort of empirical sort of estimate of the performance, and we think it is reasonable to, there's been a lot in the literature already about, the closer your inclusion-exclusion criteria are to a target trial, the universe, the sort of gap between what is found in non-interventional designs and what is found in trials reduces. And so we want to get a sense with a lot of replication activities, lots of different groups and even different approaches, where that gap is. And I'll cheat here and say the two of us were on a panel like days ago, and so I'll throw your words back at you. And
no, that's not fair.
And Dave described, we may be in a situation where we're asymptotically approaching, but maybe don't exactly get to the same place. And I think that's what this is about, So I really rely on your words, Dave. That asymptotic approach, you can still learn from that, even if it's not exactly identical. And then, like Barbara said, you can potentially open up the aperture for other types of questions that are different from trials and may not have FDA application.
I'm never giving
thanks.
up
And
again.
I think just one more thing to add to that, because this has come up time and time again in the Duke-Margolis discussions as well. And that's like, what's the role of replicating clinical? You bring up a really good point that doesn't that sort of the value of real-world evidence is not to be as controlled and have as low external validity as randomized controlled trials. And the way I look at it is it's almost like if you can do one thing well, then maybe you can do some other things. And the concept is, well, if you can suspend your view of real-world evidence for the moment and say, within this data environment or this database, can you restrict look at the very narrow population that might have been looked at in the clinical trials and try it as best you you can to define things exactly the way it was in the trial. If you can sort of get close or replicate that result, then at least you might be doing something right in terms of being able to generate effect estimates. And the sense is that if you can do that, then maybe, once you expand that aperture and actually take a more real-world view of the population and what you can answer, that you might have a little bit more confidence in those subsequent results because you've demonstrated as a research group or as a database that when you have to, in that particular case, you could get close to what the clinical trial said and then sort of back away and say, okay, now what can we really look at now that we have a more routine population? Right, okay. Good, and maybe it's part of the certification? No, okay. We won't go back to that, but thanks, Barbara and David, for your comments. We're going to roll right into our final session of the day, which, as we've discussed, we will be looking at over the next year FDA to publish their draft real-world evidence framework, and this meeting, our third annual real-world evidence meeting, will be about this time next year, and we'd like to talk about sort of, to keep us on track, what should we be talking about a year from now? What would we think when you say, wow, over the last year we've made a lot of progress? So I'd like to invite our panelists for this next session up here. And I've already been forewarned, they might go a little bit further out than 1 year in terms of what should we be doing, which is okay. Joining me up on stage will be Marcus Wilson, President of HealthCore. Joe Selby, Executive Director of the Patient-Centered Outcomes Research Institute. Hi, Joe. And Rich Mashicki, Chief Medical Officer at PhRMA, the Pharmaceutical Research and Manufacturers of America. And I think that Rich has some slides. So Rich, we'll turn to you first, if you're amenable to that, and then go from there. Thanks.
Well, knowing that it's totally hopeless for me at my age to be able to read any of that up there, I have turned to bringing a few of my slides with me. So I want to begin again by thanking the agency and Duke Margolis for putting this together. Obviously, an extremely important effort as we look at how to generate the right evidence at the right time with the right drugs. And we all believe that there is great promise in real-world evidence. I'm glad to see it's the same slide. PhRMA itself has been very involved in the thought processes around real-world evidence and played a significant role in outlining the deliverables that are part of 21st Century Cures and PDUFA 6. And when I say PhRMA, I also mean our member companies who have played a huge role. We have also put together a working group based on this, representing a large number of our member companies. And I have to say, they put together a points to consideration paper, which in fact, after reading this weekend, the Duke Margolis paper, I saw a tremendous overlap and a real synchronization, which in the end didn't surprise me since many of members of that working group were also members of our working group. So I'm pleased to say that seemed to have some coherence in our thought processes. So if we look at this timeline, we see that by the end of this year, we all hope to see a framework created from FDA on exactly how we're going to begin to think about this, and so meetings like this greatly inform that. So what does 2019 bring us? Well, in fact, we go to experience, and hopefully all of these different things that we've been hearing about in the last session will begin to show us some light, begin to give us some real life, real world, if you will, experience with real world data, and begin to help us move towards the guidances that we expect coming out in the future that have been outlined under those legislative initiatives. Now, we've talked about in this meeting and other meetings about the various uses of real-world data and its transformation to real-world evidence. but we're obviously going to focus on the boxes that are on this slide in red, which are those that probably have the primary regulatory framework. And, we've grown used to the idea that we can use it in safety analyses. And FDA has certainly led the way in doing so with the Sentinel initiative. But at the same time, the outside of that framework, only in rare diseases have we really used it to begin to assess efficacy. So we really still need this kind of regulatory framework. And as we have thought, and I'll even say as I have thought, about where do we need to go in general in order to make this real. So I think there are 5 things that the future needs to hold for us as we move forward. Of course, it begins with clarity. And that's what we've been talking about, the framework and the guidances that sponsors are going to need from FDA in order to achieve this. The second we've been talking about as real experience, right? So that's the pilot studies, the other studies that FDA has inaugurated, and our own efforts at comparisons that we've just talked about from Barbara's discussion. But if we're really going to make this real, then we also need to talk about interoperability as a very key element so that we've all said the more data, the better we feel about the results we might be able to garner from that data, and that will require interoperability in order to achieve that, and that will require agreement on what those standards should be for the ability to pool or link the data sets. And then we get to acceptance. We can do all of this, and if we don't actually make this a real part of drug development, if this becomes a one-off or used just here and there, I really don't think we've accomplished all that much. So it really has to be something that the FDA review divisions think of as an integral tool, as well as the sponsors think of it as an integral tool, and it becomes a routine thought in how we incorporate this into drug development paradigms. And then finally, I would argue there's integration. So if real-world evidence is in fact a tool that we've been successful in this process of making real, then if we truly want to change how drug development is accomplished, we need to also look at how do we combine it in drug development programs with the many other tools that are being developed, both some of them from PDUFA 6 and some of them from 21st Century Cures, as an example. So I'll take each of these apart for just a little bit. So let's start with clarity, right? And that's what we're mostly here for. So in the thought process that we've had at Pharma, we've said that these guidances that we are coming forward with should leverage our existing guidances. It should address the timing of meetings between FDA and sponsors. We also want it to be very careful that it's not overly prescriptive in its direction. So I know we're asking for clarity, but don't be too prescriptive. I understand there's a tension there. But it's flexibility that's going to be necessary, because I think we all recognize that how we then use this is going to depend greatly on the drug, the disease characteristics, and the types of data sets that might possibly be involved and available for potential use. And so that flexibility is going to be very key. And there are a number of elements up on the slide that probably need to be considered as we think about how to move towards acceptance in those particular arenas. So delivering the 21st Century Cures and PDUFA 6 guidances that have been outlined is going to be, I think, a seminal event into our future. But it's going to require the kind of thought that we're trying to give it today. So experience. You've already heard a lot about what's going on in order to begin to generate this kind of experience. And so I'm not going to belabor it in the interest of time, but rather to say that to gain this experience will continue to take, I think, two things. Courage, on one hand, courage by the sponsors and those who are willing to invest in these pilots, and courage on the side of the agency, and then time. It's going to take time from the agency and time from sponsors to have the right kind of conversations that can lead to useful pilots that can really begin to inform us. Interoperability has received a lot of interest over time. And I don't, again, want to belabor it other than to reiterate the importance of both data and system standards that we need to develop as we move into the future here. And acceptance. So I've talked about the importance that the agency broadly accepts these, not just the Office of Medical Policy, but rather it's something down in every review division. And so a couple of basic premises that, as we talk about acceptance, I think it starts with the acknowledgement of the value of real-world evidence in the regulatory decision-making. And I just want to reiterate that. Acknowledgement at the agency level of the importance of this as a tool. Acknowledgement that it has, in fact, unique advantages, and so therefore needs to have always serious consideration in a drug development program. And then I think we need to focus. Our acceptance should focus on sort of the low-hanging fruit first. As we think about 2019 and beyond, we need to focus on the approval of supplemental indications and the fulfillment of post-marketing requirements and commitments. And so, as I mentioned before, the final point that I had thought through was integration. So up here, I don't know if you can read it. I know I couldn't hardly read it. But there are a whole host of different areas that we are actively all working on and developing. And if we can just learn how to truly integrate the use of all of these into our drug development programs, I think we will make a big difference in how fast and at what cost we can get new drugs to patients in a timely fashion. Thank you.
Thanks, Rich. I'll go ahead and turn to Marcus. You have a unique perspective, and you also have been with us all day. So you heard a lot of the things that were discussed. So what do you think, from your perspective, would be we need to focus on going forward?
So I have a question actually for the audience on this, and the question is that how many, or is there anyone in here that believes that that in the current environment we have all the information we need once a product gets on the market to make all the decisions we have to make after that, whether that's payer decisions, whether that's provider decisions, or whether that's patient decisions? Is anybody who believes that? Come on, you can be brave. Courage, I heard the term. The only reason I ask is that regardless of how you feel about real-world evidence, we've got a really, really big problem in healthcare. And that is that majority of the time we don't know what to do. And it's actually at the individual level we don't know what to do more than anything. And so my real question and my challenge to myself all the time when I sort of check my own thinking on this is that if not real-world evidence, then what? You know, what's the alternative? We spend a lot of time debating whether we should use it or not and in what form we should use it. But you know, I don't think, if there's another option, then let's go down that pathway, let's explore it. But I think that, I'm kind of tired of the debate and saying whether we should or shouldn't use it. We should be using it because I don't see any other options. And so the question is, how do we harness the power of real-world data to close that gap in evidence? I spend a lot of time, HealthCore is owned by Anthem, and I spend a lot of time, Anthem spends a ton of money on closing gaps in care. And gaps in care are when you think you know what to do and you try to get people to do it, right? You hear about variability in care, reducing variability. And my debate back to them all the time, and they're beginning to actually to really listen to it, is that, don't try to control variability in a place where you don't know what the right choice is, because you may be actually limiting options to something that may not be the best options. And so that's where we're all sitting in that world, and we're all impacted by it. So we spend a lot of our time now as a company thinking about, okay, how do we change that? Because we're sitting in the middle of a world, some refer to us as a data partner, or we don't really think of it that way. We really think of it as a stakeholder in the industry that has assets that can help further this thinking, and that's what we are spending a lot of time now doing, and including to the point where we're getting past just being in a reactive mode to those who are willing to fund a pragmatic clinical trial. As an example, we have 4 currently ongoing and more in the pipeline that we're doing, but actually getting to the point where we say, you know what, maybe we should start funding our own pragmatic clinical trials and closing some of these gaps that others aren't willing to invest in closing, because at the end of the day, it's our members and our providers and us who are going to benefit from knowing that answer. And I think that from my perspective is something. So I really am tired of hearing the debate about whether we should do it or not. Let's get past that and let's figure out how to do it. And I think that's what the future needs to hold, is really a really good prescriptive model that says, okay, we're going to do this, and here's what needs to be done to do it. Because if you do that and you're the FDA, what's pharma going to do? If you say this is where the world has to go, they're going to start doing it. What's payers going to do? They're going to start doing it too. We're all going to kind of get on the boat and start saying we need to answer that question and do it and get to it together. So the only last thing I would say is that when we're talking about the OPERAN study, and I think the idea of doing the OPERAN study is a great idea, the OPERAN project, there's many studies. But the question that kept coming up to me is exactly what David said, drew our attention to on this, is that what do we expect to find? What's success in that? And I actually hope we find differences, because that's actually what, and that we embrace that difference. And really, I think, Barbara, you mentioned this, that it's, okay, let's figure out why. It's not the what, but why do we find the difference? Because that's actually where we spend a lot of our time. When we see a difference in doing a real-world analysis from what the existing data tell us, it really tells us that there's a reason that there's a difference. Is it the data? Is it the way that the data were collected, or the incompleteness of the data, or is there actually really something different about the population or the treatment setting in which they receive care? And I think that's a really important piece of this too. So let's just make sure that there's a healthy look at the OPERAN, because you could also kind of take a cynical look and say, well, we just want to prove that RWE is not useful for us, and that it's always going to find differences, and that's a death knell for it. And I don't think that's the right approach. And I don't think that's the case. But let's just make sure we got the right lens on when we're looking at this. But I'll hand it over to the expert.
Joe?
Here.
No questions?
Nope.
Okay, good. Okay, hi. Thank you, Greg, for pointing out that I didn't spend the whole day here. And
Neither did Rich.
Let me apologize for telling you what the next steps are without.
We're about the future.
Listening to
We're
anything
about
that
the
you
future
said
here.
up to this point. So it's a pleasure to be here, and I do want to say that when I came to this meeting last year, I was really grateful that the FDA made clear, and they made clear in everything they did in responding to 21st century cures, that in their world, in their minds, the question of real-world evidence had to do with the ways that previously collected data could inform both observational studies and pragmatic trials. So I still hear people today in this meeting talking as if real-world evidence was just observational data. And I felt like the paper, as beautifully written as it was, didn't say at the beginning, by the way, we're just talking about that one type of real-world evidence, that type which is based on accumulated data and nothing more. So the point is that there are two critical opportunities that real-world data present to us. And the one is to conduct analyses on data that have been previously collected with no need to contact patients, with no need to intervene with clinicians or healthcare delivery systems. And that's the so-called, really pretty much retrospective observational studies. There's another type of real-world evidence that I would venture to say comes from a different source of provider data. And that is either pragmatic randomized trials that require randomization at the individual level, or prospective observational studies that require individual consent at the individual's level. So both of those opportunities of getting individual consent open you to linking data then to any number of other sources of data at the present time or in the future, in secondary uses of the data, in pooled data. Now, I've had a lot more experience with that second type of system, which is, actually, I guess I've had some experience at Kaiser Permanente before, where they had kind of solved the problem of linkage, and they could, of linkage of EHR data and claims data, so that they could do both types of trials. But in the real world, that PCORNet and now the People-Centered Research Foundation find themselves working with, there are a lot of owners of electronic health record data that don't have access to the complete claims data or the out-of-system use. On the other hand, these systems offer, for the first time, very strong linkages to the delivery systems from which those data come and ability to access patients. So on the one hand, you've got perhaps closed systems of one sort or another where the data are already practically linked, claims and EHR data, and on the other, you've got systems which for the first time have given us linkage to the systems and the providers. And if there's one thing we've learned at PCORI is that if somebody who's proposing to do a pragmatic real-world clinical trial, if there's one thing that we need to double check before we fund, it's that they actually do have the buy-in of those systems and the clinicians in those systems. Without that, the best designed pragmatic trial falls on its face. So a couple things. I want to make clear that this is not really just an American problem. We have probably more diversity in our administrative, we probably have more diversity in things like our electronic health record data. We may have more similarity in a way in our claims data. I had the real pleasure a couple weeks ago of being in Canada, where they are getting ready to build a national data platform, and they're wrestling with all manner of issues about whether we should actually go for electronic health record data in a big way, recognizing how costly it is, what an unexplored territory it is to looking at claims data, how much more heterogeneity there is across systems in this electronic health record data. But on the other hand, recognizing that without it, as Rich said in the leadoff, we'll be feeling pretty good about measuring safety and not very good about measuring comparative effectiveness. So PCORNet, as an example, is an example of a network that really aimed to go for the electronic health record data and beginning to standardize that. And I think that is essential. The second thing is I want to just take a minute to describe a new traineeship which PCORI and the Agency for Healthcare Research and Quality just announced the first 11 sites that will be funded to train researchers based in these healthcare delivery systems. So I am really hopeful that we will make some progress over the next few years as we train more and more scientists, particularly to work within healthcare delivery systems, but scientists who can appreciate and make progress in getting data out of electronic health records in a standardized way, maybe even make progress in standardizing the data that goes into electronic health records. That would be great, and also begin to break down some of these barriers between systems and holders of administrative data, so that in a greater number of sites, we can look for that kind of linkage. So in closing, what would I like to see in 12 months? I'd like to say that we've made some progress in both types of linkage. That is linkage of electronic health record data and claims data on the one hand, and I think we do that by, among other things, building the demand for greater linkage, building the demand for this kind of research. And that takes in turn heightening the appreciation of what this data can do. What it's already done, there are many examples in the literature of major contributions that were achieved by linking EHR and claims data. So heightening that, and also heightening the understanding of the actual risks and the security mechanisms that can be put into place to protect privacy and data security. And on the other type of linkage, the linkage of clinical and administrative claims data with the providers of healthcare. And I think maybe even more the frontline clinicians than the CEOs of delivery systems, but both. That would be an absolutely fabulous step in realizing the potential for the concept of learning health systems. I'll also just weigh in on this comparison of trials versus observational findings for the same question. I appreciate that you could find 99 observational studies that agreed with the trials, and you'd still have to be just as suspect about the 100th, I think. But I think there's a ton to be learned about observational studies from comparing them one at a time to trials. So I'm in favor of that. And the last thing is that when you can reach patients using real-world data, you can begin thinking about capturing some of those outcomes, those person-reported outcomes that David was talking about, that you can't do when you just have EHR and claims data. So thanks.
Okay, great, Joe. Any other comments based on what Joe said? No. Okay, so I'm going to ask a question. It's like, who's going to lead here, the federal government or the private sector? And we've heard so much today that FDA is doing a ton, and they've got all of these projects that David outlined, so many more than I think many of us were even aware of, and it's so exciting to see how much is being made there. And we know that the agency is coming out with this framework by the end of the day. But I suspect that we're not just waiting for that framework to come out to say, okay, what should we do? So many other groups are doing things to replicate trials. We heard a lot today about exciting things that are happening in pragmatic trials or bringing randomization to the clinical setting. We also heard a lot about, in terms of mapping to regulatory thresholds, maybe a pragmatic design, it's a little bit easier to think of how that might be adequate and well-controlled, or how it might reach the substantial evidence standard. Much less is known how an observational study might meet the adequate and well-controlled standard and get to substantial evidence. But from your perspectives, it seems like, where do you think the progress really needs the most work over the next year, over the next couple of years? Is it on improving the data quality? Is it on making sure that our research studies are adequate and well-controlled in terms of methods and analytic techniques? Or is it on doing more randomization and making that easier to do in the setting? Or is it, I think the answer, or is it all three? And what's your, we have a few minutes left, but what's your, from each of your perspectives, what should the real emphasis be in the short term over the next year or 18 months?
Well, I'm going to pick up on something you said at the very beginning about, is this responsibility of the government or some other agencies? I don't know, maybe like PCORI, but this is just an immensely daunting challenge. All three of those are critically important, and I think coordination among efforts and collaboration between efforts has got to be the challenge and the only way forward. So Sentinel is for example, a remarkably large, high-quality collaboration that's been going on for 10 years, I believe, and certainly has made major progress and contributions based largely on claims data. So I think that these efforts to look at linking Sentinel with sources of clinical data, for example, PCORNet, they make all the sense in the world.
So I think in terms of it takes a village, I think it takes an ecosystem here, and I think it means all the players have to step up to the plate. And we have to address this in all three of the directions with all of the players that you've outlined in order to achieve the progress. I'll come back to that need for combining in a sort of iterative process, right? What we're going to learn influences how we provide the right guidance and how we gain acceptance. It all works together from all of those directions. Now, I mentioned courage in my previous comments, and I think partly that comes back to the idea of it's a huge investment for sponsors to try and give that experience, right? And they have a lot to lose, not just money. They have to lose, so if their product that showed it was worked in a randomized controlled trial fails to show a benefit in a real-world setting, what does that mean? What are payers going to think of that? How does that play itself out? So we're going to really have to think about what does it mean when there are differences? What is the truth of that? Why did this turn out different from that? And perhaps we'll learn a lot in the midst of all that. But I think that laying out a framework is this key next step. And that framework has to allow the space for sponsors, who I speak for, to be able to have that courage to step into that space, give that experience so we can have this iterative process.
I think both comments are right by Joe and Rich both. Just to build on that last point, every project we've undertaken, especially as we get into the randomized side of things and pragmatic trials, we learn so much, and most of the time it's so much about what not to do. Right, and so there's a patience that has to be built into this as we work together, regardless of who's sponsoring it, and because one of the key things that Joe said before, and I think you were getting at this, is that it's not just a matter of having a great design and a very patient and generous sponsor and all this, but you also have to have an environment in which is going to cooperate with you to do the project in the first place. So the provider community also has to be, we have to find ways to incentivize them to participate in these, because often we're taking these studies to research naive settings, or they've done them, but they've done a traditional randomized clinical trial and don't understand the differences between these. And so I think there's a learning curve that we all have to go through, and I think we have to be open during that process, and we have to be patient as we go through it. And the shared learning, I really think the 5 points you made were really good, Rich, and I think that the shared experience is the one I kind of glommed onto probably more than any of them, because as I've learned, we're willing to share that with others that we're working with. I think it's going to take all of us doing it to kind of build that foundation solid going forward.
If I could just put a slightly finer point even on what Marcus just said, it's you can have healthcare delivery systems where there are clinicians who have done research, and they've been part of huge multi-center traditional clinical trials, and they've maybe recruited 5 or 10 or maybe 20 or 25 patients. They then propose to do a pragmatic trial in the same setting, and you come along and fund it. And you're not, when you make an investment in doing a pragmatic trial and you invest in a delivery system to do it, you are not looking for 5 or 10 or 20 patients. You're looking for clinicians to be aware of the study, the delivery system to be aware of and supportive of the study, so that a large fraction of the patients who would be eligible get recruited into the study. And that's what makes it pragmatic in the sense of generalizable, and that's what makes it affordable.
Well, thanks. We're a few minutes over, so I'm going to end this session with that, and I'd like to thank the 3 panelists here for bringing forth your ideas. Okay. But stay there. So just stay seated for a moment. I'm just going to go ahead and close out the day. I'm pretty optimistic. We've covered a ton of ground today, and a ton of ground has been covered over the last year, and lots of interesting things that have been done. A lot of alignment on some of the more controversial topics I think we heard today. And Marcus, I'd say overwhelmingly from today, it's a yes on real-world evidence. I heard nobody has said, at least today, finally, that we shouldn't be doing this. There's a lot of alignment on that. And I'm pretty optimistic that while we are making tremendous amount of progress in coming up with innovative ways to reach patients in the clinical setting through apps and doing pragmatic designs within the clinical setting, no one has ruled out the possibility that observational designs can bring some useful information, and no one's ruled out that they can't potentially reach adequate and well-controlled. The work is, how do we make sure that they can? How do we make sure that the data and the methods and the uses are most appropriate? And then third, I'd say that it seems like we have all of the right organizations and all of the right people in the room. I'm blown away by the amount of progress and the energy that you all bring to this topic in a way that is complementary to FDA. I think everybody wants to help FDA and wants to help make sure that we together can get there in terms of best uses and most appropriate uses of real-world evidence. So before we adjourn, I just want to thank a few folks. First, I'd like to thank all of our panelists and speakers today for bringing their ideas, controversies, and arguments to the discussion. It was a very successful day because of that. I'd also like to thank all of the members on our advisory board and organizations in the Duke-Margolis Real-World Evidence Collaborative. We couldn't have done this without you or have made as much progress on the white paper that was released, but the others that are in development. And then lastly, in terms of our staff at the Duke-Margolis Center, I'd like to thank Christina Silcox, Jonathan Bryan, Catherine Frank, Morgan Romine, and Sarah Saprisi, and Elizabeth Murphy for all of your help along the way and helping to make today a success. So thank you again, and safe travels, and thanks for making today so successful.